Good morning, everybody, and happy solstice. Thank you for coming today. I want to remind you that the important land acknowledgement that Heather Benway made yesterday is still important today and tomorrow and all our days here. Today, we're going to have an all-day session, our evolving understanding of the biological carbon pump. On the Science Steering Committee, we started discussing aspects of anthropogenic impacts on the biological carbon pump, and it became very clear how limited our understanding is. And Dr. McKinley said it very eloquently yesterday, our uncertainties are just so large. So ultimately, we decided that we would go ahead and have an all-day session on the biological carbon pump. This morning, you will see, one second, I should have pointed out this beautiful image is by Kristen Karmhard. She's a postdoc at NCAR, and she's also an aspiring science illustrator. So this beautiful image, please recognize that. This morning, we're having overview presentations to get us all on the same page and on a lot of the knowns of the biological carbon pump. Then this afternoon, we have spotlight presentations that are shorter, and they're going to focus on some more recent insights that might stress some of the ephemeral nature of the biological pump or new mechanisms of getting carbon, organic carbon, out of the surface ocean. And then we're going to look to the community, both online and present here at OCB, to work together. If you're here in residence, we're going to meet outside and have community discussions and community input on various topics, and we'll give you an overview of that. If you're participating online, Tristan and Seth Bushinsky, a co-organizer of the session, will have an online option for you to meet. And I'm looking at Emily Osborne, who's the co-conspirator, and if you ever want to organize a session, you should try to get her on board because it's amazing to work together. And with that, we're going to welcome our first speaker, Dave Siegel, with a truly inspired talk and title.
Truly inspired. Truly. Oh, sure. I'm not planning on doing any gymnastics, if that's what you mean. Okay, my task is to introduce the biological pump and to talk about the exports project. So, title is The Ocean's Biological Pump, the Pump Pathways. Oh, I need this, don't I? And the 3 pump pathways in a field campaign to bind them all. I've been told not to do any Lord of the Ring jokes. I've been told to do a million Lord of the Ring jokes. We'll see what happens. Okay, so with that, let's just talk about the biological pump. It is the 2 biological pumps, the physical pump and the biological pump take carbon from the surface ocean and put it down to depth where it's sequestered for months or sequestered for millennia, depending upon where it gets to. The physical pump is pretty straightforward. We know physics. We know what the air-sea exchanges. We know what air-sea exchanges are. We know what that is. We have some knowledge of the DIC distribution and circulation. We can estimate that. The biological pump is a mess. We have to understand how the ecosystems work, how the food web works, how the circulation interacts with that, how that reacts with the biogeochemistry of the ocean. So we know that we need to do more. So it's a hard problem. So if we think about the biological pump, it's this coupled set of pathways that transfer organic matter to depth, and it's a pathway for rapid carbon sequestration, of taking carbon from the surface ocean down to depth. That creates that vertical gradient in DIC that what Galen and everybody talked about yesterday. And that in turn helps drive the physical pump, letting more CO2 evade into the ocean, or invade, I guess. If you turn it off, if you can create magic and turn it off like you can in the model, the atmospheric CO2 rises greatly. But our estimates of export and our estimates of sequestration are horrible. We need to do better. So if you think about it, what are the processes? We fix carbon through photosynthesis. The animals in the ocean eat that photosynthesis, those photosynthetic products, making suspended and sinking particulate organic carbon, making DOC, making dissolved organic carbon, creating a food web that spins around. Some of those materials are sink or, here we go. Some of those materials are exported into the twilight zone, the area beneath the euphotic zone where these materials are consumed. The rate at which that export happens and its consumption below that determines what that remineralization scale, that determines how long, that and the circulation determine how long things are sequestered for. So if we think about it, there are three basic pumps that we need to kind of deal with to understand, and they're all important enough that we have to do it. So the first is the obvious one, it's sinking particles. We've been studying this for 100 years. And that is where we have sinking aggregates, fecal matter, or whole sinking phytoplankton cells that go from the surface ocean and sink slowly or fast through the ocean, depending on how they're packaged. The other pumps are the diode vertical migration. This is where we have fish and zooplankton that eat organic matter in the surface ocean during the nighttime, go down to depth to avoid predation, allegedly, since we don't know really what they want to do. But we believe that that's what happens. Nobody's done an interview with a migrating thing. Anyways, and then they respire, they poop, they pee, and do all the things that animals do at depth, rapidly transporting organic matter from the surface ocean down to depths greater than 500 meters in a day. And the last is this mixing pump. This is where suspended particulate organic carbon and dissolved organic carbon get pushed down to depth by either mixing processes as the mixed layer evolves, or in the overturning circulation of the global circulation. So I'm going to talk about these sort of things, talking about this in terms of a recent model that Michael Nowicki. Michael, raise your hand and say hi. I told you I was going to embarrass you. You know that. And so Michael's a graduate student of Tim DeVries and myself at UCSB, and he's developed a steady-state, data-constrained 3D model of the biological pump, including all three of the pumps. It has an ecosystem model with two phytoplankton, two zooplankton. It uses satellite data from net prime production and the particle size distribution as a boundary condition at the surface. It then assimilates phytoplankton carbon, zooplankton distributions from data archives, the global DOC distributions from the repeat hydrography, oxygen distributions from everywhere. And he's done it with a bunch of ensembles, so we both get a mean state and then the uncertainty about that state. So we look at what the result of this, and this is just an global mean, just to get a sense of what the relative proportions of the three pumps are. We can see that the total export is something like 10 petagrams carbon per year from this model. Most of it is happening due to the gravitational pump or the sinking pump of particles into the interior, and most of that is by fecal matter in this particular model simulation. 20% of it is due to the mixing pump, and 10% of it is due to the migrant pump in this depiction. If we look at sequestration, okay, so sequestration here is measured, and there's many ways of doing this, but it is measured here as the inventory of remineralized carbon that remains from contact with the surface ocean, in this case being in this steady state model. So well, what are we saying? So we have all these pumps that are pushing things down, organic matter down to depth. That organic matter turns back into DIC. We're looking at what is that total inventory of DIC that is in this. And from these calculations, the total sequestration is about 1,300 petagrams of carbon of remineralized organic matter that is sequestered due to the biological pump. The turnover time of that, if you take that inventory divided by that export flux, you get a turnover time of about 127 years. Here, 80% of it is due to the gravitational pump. Okay, again, most of it due to fecal materials. Only 8% of the sequestration is due to mixing. And that turnover time is in this case, is just a couple decades, 54 years on a global basis. If we look at, well, the migrant pump sequesters about 12%, so it's with a much longer time scale because those materials go deeper into the water column than the mixing, which happens on much more shallower scales. So here's just an example. This is, I'm thinking one of the ways as we improve to do these sorts of calculations to constrain what is the global impact of the biological pump is these global inverse models are a good way to do this. And this is one example of how to do this. This is not the only way to do this. There's lots of assumptions in how all the models are built, and that's the important thing to understand next. It does account for all three of the biological pathways separately. But improvements are needed. This is a steady state model. We need to have time dependence, both seasonal cycle as well as interannual variability, as we all know. We need more and better complete data sets to drive these things. I think the future, and I'll show some examples in just a little bit from export, but having some global assessments of zooplankton, of large particle sizes, those sort of things will really help. And the parameterization of processes need to be improved. There are assumptions that are applied that says that this process works like this way, and we're going to parameterize it the same darn way everywhere. And that may not make some sense. And there's some ways that there are definite improvements to be made. So we need a field program to do all this and to constrain the three pathways and all that sort of stuff. So this is basically, can I do this? There we go. This is basically why the exports project exists. Okay, the goal of exports is to predict the export fate and carbon cycle impacts of ocean net primary production from satellite observations. You think about it, NASA, and it's largely driven by NASA. It's a NASA field campaign officially. I am the science lead of the exports project still, and if you think about it, NASA has invested decades into understanding net primary production. Net primary production tells you what? It tells you how much carbon goes into the food web. It doesn't tell you anything about carbon cycle. So what we're trying to do is help bridge that, especially as NASA's launching the PACE mission that's coming up shortly. So the focus here on exports is to quantify the pathways and the net primary production fates over a range of ecosystem states. So we conducted 2 field programs in a highly recycled area and a highly not recycled area. And I'll show that in a sec. The idea is to emphasize a predictive understanding by measuring and modeling the regulating processes, and it leverages a bunch of stuff. Advances in remote sensing, ocean optics, particle imaging, genomics, machine learning. I mean, it's a good time to do this sort of stuff. Autonomous vehicles. So, everything that we need to do, we need to start from science questions. Why are we here? What are we doing? And I think it's very important to always remind yourselves of what what we're trying to get at here. We've designed exports with 3 basic questions. How does the upper ocean ecosystem export material from the surface into the aphotic zone, into the interior? What happens to that material? Those 2 things together to say how much organic carbon has answered these questions, tell us how much organic carbon came from the surface, and how quickly does it get remineralized as we move into the interior. The last question is, how do we use this knowledge to reduce the uncertainties in contemporary, so it's diagnosing what's happening today and future. How do we better predict these estimates of the export and fates in that prime production energy? So where are we? Exports was designed to be in 2 phases. You can sort of think about the first phase, which is the field phase, which is aimed at getting at the first 2 questions. What leaves the surface ocean and what happens to it once it gets out of the surface ocean? We're focusing on pathways. We've created these sort of sets of wiring diagrams, as you can see here, to sort of talk about how the food web works, how that interacts with the carbon cycle. We link, we're largely, largely a NASA-driven project with contributions from the National Science Foundation and other agencies, and the National Ocean Center, by the way. I'll get to that in a sec. And so, everything we do, we want to link. We want it to be remotely sensible. In every way that joke works, So it has to be useful, but it also has to be able to link to satellite missions or satellite data products. We had 2 cruises, one to the Pacific that I'll talk about very briefly, and one about the Atlantic that we did just over a year ago, which is kind of exciting, and we want to focus on that. With the sampling program, I'll talk about had both multiple ships, lots of AUVs, we were very lucky to have a dedicated data management team. We could not be anywhere close to where we were without having people who are making certain that we actually do what we're supposed to do and get data into the databases. It's it's both carrots and sticks are involved. And we've spent a lot of time documenting protocols, and there's been a NASA technical memorandum. Every measurement we have a protocol for. Everything's shared, everything's public. So with that, the experimental approach has 2 ships. One acting as a process ship, measuring rates, following a Lagrangian float, which is this thing here, that's sitting at the base of the episodic zone, moving around, giving us a center to be able to do a kind of quasi-Lagrangian. It's not completely Lagrangian, but as good of a Lagrangian experiment as we can do. The idea is that when we're making rate measurements, we want to be making measurements of the same stocks and the same rates as we're following along. The second ship is a survey ship that is measuring around things, doing other experiments, intercalibrating between all the different robots, and every sort of robot that we can imagine putting into the water column, we put into the water column. Okay. We have a detailed autonomous program as I described. The idea is to get assets out there before we go to the site, so we know what the environment looks like. The ideas are to look at sort of ranges of scales with floats, long scales, with detailed spatial measurements from the gliders, and this little Lagrangian float is an experimental reference center. So our first experiment, and this is the only slide I'll have on that, was in the Pacific, where papers are coming out now on that. It was done in August of September 2018. It's a low biomass, low rate, low flux environment. So you can sort of think it is it's one end of the spectrum of export and export efficiency. We found a highly recycled food web. I think Heather McNair will talk about that in one of the lightning talks. It was not quite a steady state, but almost. We could see differences in things, and it gave us a lot of confidence that we can see careful small changes in data sets, in coupled data sets, which was kind of exciting. It had very low inductions of the physical organic carbon pump. The physical mixing pump was pretty small, at least during the period of time we were out there. We did have, Debbie will talk about the role of SALPs in the system, I think, right? Yeah, good. Okay, and yeah, I thought so. But, so a lot of the thinking flux were largely driven by that, and we have a special issue of Elementa coming out. The Atlantic, we had 3 ships. So in the middle, there is the Discovery, which was the survey ship. The far ship is the James Cook, which was the process ship. And in the foreground is the Sarmio de Gamboa, which was the Woods Hole Oceanographic Ocean Twilight Zone experiment. So they nested with us in the Atlantic. The ships at the National Ocean Center were amazing. It was a wonderful experience. I think everybody who sailed will agree with that. I think we've ruined a whole generation of oceanographers, by the way, for funerals. What else do I want to say? It looked like this the entire time. Okay, so before we went out there, we went to the PAP site. We chose a place to do the experiment in a anticyclone. We spent an awful lot of time figuring out how to do this and why to do this. I like to thank Zach Erickson, who's here, was part of that team. But we had a team on land that helped drive where we were. We had 2 gliders out. The Slocum glider from National Ocean Center. Felipe's here someplace, right? That was her glider. And then SG is a sea glider from the University of Washington, from Craig Lee's lab. And what we did is we had these things out there. We're surveying things as quick as we can. The 2 ships that are shown are in orange and yellow. You can see us running away for a while, and we're running away. I'll talk about that in a sec. And we put out most of, thousands and thousands of, not thousands, 47, 40 some odd things were thrown in the water that we tracked in real time. This display was on the bridges of all 3 ships. There was, this detailed shore to shore of the ship and back to ship data sharing was fantastic. It actually worked out really well. So let's just talk about the cruise. There's a rain joke in, never mind. Anti-cyclone A2 was what we chose. It remained coherent throughout the experiment and retentive. So that meant that the Lagrangian float stayed in the center of that. And that helped us figure out where the center is. By doing ADCP work and all that sort of stuff around it, we actually knew where the center was at all times. And that's how we're kind of, that's one way of looking at this. We ended up with we have some evidence of some overwashing during the first storm, but for a large degree, it was the Lagrangian experiment that worked out well. Weather was an issue. Great Britain had its greatest wind power generation during the month of May last year, and I know why. So that's the Soren. No, it's another bad World of Rings joke. So this is the North Atlantic. That is us. We chose the best place to go. So we had 4 major storm events that are very important, I think, about how the evolution of the particle fields worked here. We did lose 7 days of sampling. We did have 7-meter seas, and thank God I was on the Discovery and not on the Sarmiento. Sorry, Ken. Where am I going? Okay, so when we got there, what was our goal? Our goal was to look at the demise of the spring diatom bloom. We got there as we got there when the ocean was chock full of diatoms and large diatoms, and and the silicate concentrations were really, really low. So we knew that we got there as the bloom was fully developed. In time, both the community shifted. This is the first page of the dashboard from the IFCB, which is a high throughput microscope that images pictures of things that fluoresce. And you can sort of see we moved the smaller things and we moved to much more mixed assemblages. We have ways of then taking those data sets, those data from the IFCB, and to convert it into carbon. You can see initially we had a lot of diatoms and some dinoflagellates. We're probably running away here. We were able to, the underway sieve systems were able to function even in high seas, as their NOAA was kind of a miracle. But you can see that that came back up. We so we had large changes in phytoplankton biomass in time. The zooplankton, which is shown here, these are day-night tows from mock nest tows. One example showing the day-night differences, showing large amounts of migrant biomass that's being worked up into migrant fluxes now. And we have these large pelagic snails or pteropods, which and abundant, but I guess not a big part of what the carbon export was, which was kind of surprising. Okay, it's a remote sensing project just showing that we could actually see the ocean every once in a while. When we had these storms, it was pretty clear, which is just weird. It was nice and sunny days and blowing stink, but you could see that. So the feature, you can sort of see the feature where it's kind of spinning around. Below, you can sort of see that there's water's being entrained here on the 13th, where we see this little mushroom-like thing. But we're able to what to follow this in time. We made every sort of optical measurement known to man. These are just 2 examples. This is the particle size distribution of nanometer-sized particles. So this is a micron here. Okay, so these are all smaller, using a Brownian motion machine to try to get at what is kind of a novel measurement made by Xiaodang Zhang's group. And these are just measurements of the reflectance measured on a bow-mounted radiometer system, looking at the change in the reflectance data sets. These data will be very useful to help pace and its calibration and related things to that sort of stuff. In terms of production shown here are Size fractionated net prior production and net prior production, new production measurements from C13 and N15 data from Meredith. Meredith, say hi. Thank you. And, you know, the big story here is that the production rates were high. It was initially dominated by things larger than 5 microns, then coming about equal proportions as the time went on. The new production was almost balanced with the net prime production with very small amounts of regenerated production. So it's very much new. We've got some things to figure out. The silica uptake rates were very small, which doesn't. So we're still trying to figure out how all this fits together. What's shown on the right here is dissolved organic carbon bacterial production and bacterial biomass measurements. You see that they increase in time. All three of these increase in time, especially below the mixed layer in this area down here. I'll talk about that in a sec. We look at the particles. What's shown on the bottom here is we have these underwater vision profilers. These are devices that are made to take pictures of an area about this big and to work out what the particle size distribution is. This is turned into biovolume, so it's just a particle size distribution multiplied times d cubed. You can sort of think about it, but it's a way of sort of looking at what's the dominant particle sizes and particle volumes and how they change. And you can see initially we have particles. So this is 1 millimeter here. This is a 1/10 of a millimeter. So we have particles are sitting in the mixed layer here. They're of this scale, smaller than a millimeter. They stay that way for a while. Okay, this is probably due to weather, probably due to breakage. We think it's probably due to breakage. We're working on Adrian Bird and myself and others working the modeling. By the 14th, you can see that we start to have large particles. These are sort of 2 or 3 millimeter in size. These grow, these disappear. I think that's aggregation, right? And you start to see something beneath the mixed layer. By the 21st, it's here. By the 25th, it's there. If you look at Elisa Robinelli, say hi, Elisa. Thank you. Led the marine snowcatcher work. Well, the marine snowcatcher is a 100-liter bottle that you're able to look at different sinking fractions by extracting material at different depths. At the bottom is a tray, and in the tray they can collect individual aggregates. Initially, we saw nothing, and then this is just counting the number of aggregates per liter in that bottle. You can sort of see that, as the experiment moved on, we started to see larger particles. These are things that you can see and pipette out. Okay, we look at the sinking particle fluxes. I'm doing this to everyone, Meg. Where are you? Okay, Meg Estappa's group ran the trap fluxes here. The trap PSC numbers are here showing the first 2 epochs, so that's periods of time. Here and here in time, at the first part of the experiment, the flux shows a decrease of depth, but it's not very strong. By the 3rd epoch, it gets much larger. And you can see this in the gel traps that Colleen Durkin was doing. Colleen, you have to raise your hand. I'm making everybody do it. Sorry, I'm going to pay for this in some sort of way in the future. I think I owe everybody drinks. We can see that there's an increase in the amount of schmutz that was a technical term. What we see in time. We look at the same sort of thing we can see in the thorium flux data from Sam Clevenger in Ken's group. We also see this in some novel proxies. I think you're going to talk about this, right? And Colleen will talk about these data sets and how that works later. We did an edge experiment. So what does that mean? So the process ship was working at the 80 standard doing rate measurements. The survey ship was going all around finding robots, trying to intercalibrate them. We also did an experiment with the Sarmiento. The Sarmiento was there. It had a Mach 10, so it looked at larger things that we could see with a Mach net. It also had this device here, the Stingray, which had a shadow graph system on it, so that they can look at larger and identify larger things. One of the experiments that we did is we were looking at things on the edges from remote sensing. We could detect were places where there's lots of sub-mesoscale energy. We found these fronts repeatedly using both gliders and underway CTDs towed behind the Discovery. I have no idea what time this is. That's what you're doing. Good, perfect. Thanks, May. And so we towed. So Heidi Sossack's group towed the shadow graph system up and down here. We're able to identify different types of water masses. We found this subducted feature here that had high chlorophyll. It also had, and then behind after the Sarmiento, the Discovery went and poked holes in the water and collected water out of places that we got data in real time. So we transferred data from ship to ship in real time. And we're able to do that. We found these subducted water masses. There was elevated chlorophyll, as you can see here, but also anomalous nutrient ratios. We could see different microbial communities. And we could see, and the shadow graph system showed very large aggregates that are falling along on this thing. So, Zach, raise your hand. Thank you. Zach has a poster talking about that here. So, just the last thing. Just remember what happened. We did this in May. Okay. Find something that's here. I mean, the thing I want to emphasize. We got 54 people onto ships, and how many from the Sarmiento? Okay, so what is the number? 70-some-odd people from the U.S. and other places onto ships, healthy, safe, you know, in as ways we could. Ivana, raise your hand. Ivana did it was a miracle. I mean, I can't just say that. I mean, it's just I'm amazed. I have post-traumatic. I can't even say the words. Post-traumatic stress disorder. You know, I'm sure we all did. So just to wrap up, the North Atlantic experiment worked out much better than I don't think any of us anticipated. We went to go do something. It happened the way it did. Not everything was perfect. Not everything ever is perfect. But you know, the fact that we had this planning, that we had a detailed situational awareness plan. A lot of this stuff is going to be coming out and published, even the logistics of how we did some of this, because we had a team on land that was telling us where to go. We were telling the team on land where to go from us at sea, but that's in the story. We went when we saw we should be able to see this. What was interesting is we had the story behind this is we have the science story is we have this large lag between the bloom that happened early May or late April and us seeing exports happening in late May. And so what in the world is going on here? And there's interactions between the weather and advection, but also all these particle processes of aggregation, disaggregation, solubilization. All these things are going to be important. And it raises the question in my mind is like how important, JGOFS was built on these sort of production flux experiments. And really, what are we seeing? What are we seeing? So with that, Laura talked about phase 2. Hopefully it's going to happen. Everybody talk to Laura and tell her how important it is. So with that, I just want to say thank you. We had a lot of birthdays. You want this?
Thank you, everybody. Next up, we're going to get a modeling perspective on the biological carbon pump from Charlotte Laufkötter.
Can you all hear me and see me? Yes? Okay, great. Hey, everyone. Thank you so much for having me. This is my first international travel since COVID, so I'm super excited to meet you. Yes, I will speak about the modeling perspective on the biological pump. Specifically, I want to say what models suggest for the present day budget, what changes we expect with future global warming, and also the uncertainties associated with these projections. You can also see. So I wanted to start with a very quick overview of the components of the biological pump, because this is how I structured the talk. So the first one is the thinking of particles of organic matter. And David already explained everything so well, so I'll be super quick. So we have the ecosystem in the upper ocean, and there are a lot of interactions, and this resides in a spectrum of particles of organic matter. Some of them are heavier than water, so they stink. This thinking of organic matter is the main component of the biological pump. It's included in all models that I'm aware of. And it's assumed to be about 80% of the total carbon. And then the particles that are not heavier than water remain suspended in the water column, but they may still get mixed into the deep ocean if the surrounding water gets mixed down. This is the export of dissolved organic matter. It's supposed to be about 20% of the total carbon export, and it's also included in all models that I know. And then there is the joker, that's the zooplankton behavior. That has only recently been highlighted for observations. As a result, it is not included in any models that were used for the IPCC projection. In fact, there are very few models at all who try to estimate a present-day budget. So today I want to go through all these compartments and give you an overview of how big the present-day budget is, what models suggest for the changes, and a bit also on how it is actually implemented so you better understand the uncertainties associated with it. Also, I realized I have a lot to say about part 1, the thinking particles, but then much less about 2 and 3. So if in 20 minutes I still talk about part 1, don't worry, this is part of the plan, really quickly. Right, before I do all that, I wanted to quickly introduce what I mean when I say model, because this is a word with many different meanings. So most of the time today, I will speak about Earth system models. These are the ones, for instance, used for the IPCC projection. The Earth system model basically approximates the world with a giant grid, and for each of the grid cells, a large set of equations is solved that describe the physical and biogeochemical state of the grid cell. And then all grid cells together represent the atmosphere, ocean, land, and ice. And with Earth system models, we can calculate projections into the future. So these are our main tool to understand the implications of future global warming. But Earth system models also simulate the biological pump, and they do so in full interaction with the other components of the climate system. So for instance, if there's a change in the deep ocean particle flux in an Earth system model, this will affect the surface ocean, and it will also affect the atmosphere. Unfortunately, Earth system models are computationally very expensive, and this often leads to difficult decisions. So what processes do we have to include because they're relevant and we need them to understand, and which ones are maybe possible to omit and we will still understand the big picture? Every few years, there are large comparison projects for Earth system models called CMIP. The current one is CMIP 6. So if I say CMIP 6 model today, this means this is the most advanced set of Earth system models that we have. Currently, 19 models submitted data to CMIP 6. In contrast to Earth system models, I will also sometimes show satellite-based estimates today. Now, a satellite obviously is not a model. A satellite measures things. But satellites can only measure the surface ocean. So the satellite-based carbon estimates from the deep ocean are not observations. In fact, smart people have come up with algorithms that connect quantities the satellite can measure, like temperature and ocean color, with the observed carbon flux at depth. And these algorithms are either statistical models or food web models. So the carbon export from satellite is also a model estimate. A very important difference to Earth system models is that satellite models do not have feedbacks. If I calculate a different carbon flux in the deep ocean, the algorithm will not check how this will affect the surface ocean. On the plus side, these models are computationally cheaper. This allows us to have a very high spatial resolution. And obviously, they start out with something that's observed, so that's of course a huge plus. And the last thing that will pop up today are inverse model estimates. Here, the idea is that we use the observed nutrient distribution, which we know much better than the particle flux. We combine it with an ocean circulation model, and then infer how big the particle flux must have been to match the observed nutrient distribution. Inverse models cannot be used to do future projections, but they can be very powerful in understanding the today's. Okay, this is the modeling intro, and now I will speak about the biology. Here you get an overview about the globally integrated particle export at 100-meter depth simulated by different types of models. So to the left, there are the results from 19 different Earth system models that are part of CMIP 6. Currently, they project something between 4 and 12 gigatons of carbon per year. And for comparison, I've also added a few satellite-based estimates. They also suggest something between 4 and 12 gigatons of carbon per year. But maybe it's worth mentioning that the highest bar here, the 12 gigatons, are from a study that by now is about 20 years old. The most recent estimate that I show is from David Siegel, and that's at about 6 gigatons. And then finally, there's one inverse model estimate. This is the classic one from Rainer Schlitzer, and it's suggested about 10 petagram carbon per year. Now, I also have the simulated export on a map geographically. To the left, you can see the CMIS-6 multimodal mean. And for comparison, to the right, there is a satellite-based estimate. Just to make this super clear, we cannot use the satellite to evaluate the model because the satellite is also a model estimate. But it is an independent way of estimating the carbon export. And if the patterns of the two match, this can increase our trust in what the models are saying. And I personally think they actually do match pretty well. So models currently agree that there is a high particle flux in the equatorial upwelling regions, also along the eastern boundary regions, and there is a low export flux in the subtropical gyres. The regions where models most strongly disagree is the Southern Ocean. So you can see that the satellite-based estimate, but also several of the CMIS-6 models suggest a very low export flux, but then others have a really high export flux there. And it's, I think, not very surprising because this is also the region where we have almost zero observations. Right. I was going to say that for climate change, it's really important what happens to those particles after they sink through 100 meters. Because only if they really reach the deep ocean, they are prevented to exchange with the atmosphere. So therefore, I wanted to show depth profiles. So here you see depth profiles of the carbon flux in different models. The y-axis is depth, the x-axis is the particle flux normalized to 100 meters, and the line represents a different model. This is showing CMIS-5 data because I don't have the same figures for CMIS-6, but I doubt it would look much different. So you also see little stars. These are sediment trap particle flux observations, and this is a vertex station, so we are somewhere in the North Pacific. I'm showing this so you get an idea of how much the particle flux differs in different models, but also that you see how much it actually differs in the observations. So as a modeler, if one of those lines is my model, I'd more or less be okay with the model data fit. It's simply not very clear how exactly the particle flux looks like in the intermediate depth. The models here represent annual means, and the sediment trap also represents longer-term averages. I honestly have very little understanding of how the seasonality of the particle flux should look like, or any form of temporal variability. And also, we would need this type of evaluation for many different regions of the Earth in order to robustly evaluate a model. And a few years ago, I would try to collect sediment trap observations of the particle flux from different depths in order to do so. And on the map to the left, you can see the amount of stations that I can up with. The figure is now a few years old, so maybe there are a few more stations now, but this is what modelers had at the time to evaluate the models against. I also wanted to highlight that there is almost nothing in the Southern Ocean, so really no surprise we have such disagreement in that area. Here are just a few more profiles, just for you to get a feeling how well models match the data. There are some sites where the fit is pretty good. These are usually from the North Pacific because that's what the models were originally fitted to. And then models typically struggle above oxygen minimum zones or at really hot or cold sites. Based on this, you may already guess that there is not so much agreement on the carbon flux at depth. I'm going to show it anyway. So the following figures I've received from Jamie Wilson, and he kindly allowed me to show it today. But he's asking not to take any pictures yet. The paper is under embargo, but will come out very soon. But if you want to contact him, here are his details. So on these figures, you can see the transfer efficiency in a subset of the CMIS-6 models. Transfer efficiency is the fraction of particles that started out at 100 meters and reached the 1 kilometer depth. So the percentage of particles that survive the mesopelagic. I cannot show more models because many models didn't submit the full particle flux to CMIS-6. Many of them only submitted particle flux at 100 meter depth, so it really limits analysis. But we have those 5, and I think my main point becomes very clear. So to me, these models really simulate different planets. I see very little resemblance between those patterns. Yeah, and in defense of the models, until recently, to me, it also wasn't clear at all how this pattern was supposed to look like. There were a few attempts from the observational side to estimate how it looks like, and they came up with literally opposing patterns. So at the time, this was all equally likely to me. But there was an inverse throttling model that got published a few years ago, and I found it really nice and convincing, and so I want to highlight it today. It's from Weber et al. And they show that based on the nutrient accumulation in the deep ocean, there must be a high transfer efficiency in the high latitudes and a rather low transfer efficiency in the low latitudes. Now, inverse models represent a longer-term mean, so it's still very much possible that there is a very efficient export event in the low latitudes at a particular day. But I think on the annual average, this is roughly how it must look like. Now, you might look at this and wonder why. Why are the models so different? So I want to put in a block, like a little method section, and try to explain how the particle flux is implemented. So 19 modeling centers submitted data to CMIP6, but several of those models share the same ecosystem module. So I'm talking about 14 ecosystems. All of these models have a representation of the plankton community at the surface ocean. So there are 2 to 3 phytoplankton types and 2 to 3 sea. And in the models, particles are generated either during grazing, that's supposed to represent the Siegel pellet flux, or from phytoplankton directly, and that's supposed to represent phytoplankton aggregation. However, once in the model, basically all particles are the same. Most CMIP6 models have only one type of particle. Very few have 2 types. And these particles then start to sink. 9 models have a constant sinking speed, and in some models, the sinking speed varies with depth. And I want to highlight that the sinking speed in the model can be very different. So the slowest model, the one with the slowest sinking particle, has a sinking speed of 1 meter per day, and in the fastest, it's 100 meters per day. So this is a factor of 100 uncertainty right here. And then, obviously, the particles get remineralized. In many models, this just depends on the particle concentration. In some, it's temperature dependent, and in some, it's oxygen dependent. And this is basically it. So in the default CMIP6 model, you have one type of particle, it sinks with a constant speed, and it gets remineralized. I'm aware of 4 models that implement some form of mineral ballasting. So that means if the particle is associated with opal or calcium carbonate, it will sink faster. There's one model that attempted some form of hermetization of fragmentation. And I'm sure many of you could give me a long list of other processes that are important for the particle flux. They are not included in CMIP6. Also, with one exception, all models have a constant stoichiometry. So the carbon to nutrient ratio in the particles is usually. Now, you might wonder, how do we move forward from here? And I think one option, one important thing to do would be to go through the things that we do parameterize and see how well the parameters are constrained. So how much uncertainty do they cause in the final estimates? Don't have all the answers, of course. Also, I made a mistake. Actually, sorry, back. I wanted to observe why the models are not more complicated. I'll do this. I try to give you a few reasons why not more processes are included. So the first obvious reason, I think many of you are aware of this, is that many processes are currently so weakly constrained that it's almost impossible to include them in a model. I wanted to give an example to maybe then you can better understand. Let's assume I wanted to include particle aggregation into my model, which I'm sure it happens and I'm sure it's important. First of all, I would need several particle sizes. And I'm not really sure how many I would need. Also, it would make the model much more expensive. Then I would need to know what fraction of the particles actually aggregate and how that affects the size distribution of the particles. Ideally, I would need this not static, but in response to environmental drivers. So I would like to know, depending on turbulence and, I don't know, temperature, I'm making this up, like how many particles would aggregate. And even if I knew all that, next I would need a lot of observations in order to evaluate my new model and be able to judge if the new model with aggregation is actually better as the old one without. And this is a huge obstacle and one of the reasons why the models remain simple. There is another reason, though, and that is justice models are expensive. So you only want to put things in if you have a good feeling that this is also significantly affect the output. But in many situations, we already have a feeling that it may not. And I'm trying to explain this better here. So Earth system models have to conserve mass. We cannot make up nutrients. If you look at the figure here, the Earth system model simulates a particle flux out of the surface ocean, and that removes nutrients and carbon from the surface. And it also simulates a nutrient influx into the surface ocean. These two fluxes have to be roughly in balance. If they are not, if the particle flux is stronger than what goes into the surface, that means that the surface ocean is drained from nutrients over time, and the concentration will get lower and lower. And this will force the particle flux sooner or later to also become slower until the two fluxes are in balance again. So in this way, the export flux out of the surface is really tied to the nutrient flux into the surface ocean. And in this model, if I now add an additional export estimate, let's say I add zooplankton migration to this model, it's unlikely that I can actually increase total export. What will likely happen is that the model splits the existing flux out of the surface into the two new mechanisms, because the total export is tied to the amount that goes in. Because of that, modelers are often hesitant to put something new into the model, because it will be expensive, it will make the model slower, but it will likely not change the total carbon export. Back to what I tried to introduce before, how can we move forward from here? And I think something great to do would be to look at the parameters that we do have in the model and see how much uncertainty they cause, so that we know as a community on which one should we focus to get better model predictions. And this is a study from one of my PhD students that I supervise, and it's going to be published soon. And I also only wanted to, as a teaser, show this one figure. And so she created a model of the particle flux, a 1D model, very detailed, containing several different factors. And she did a huge set of sensitivity studies to see, change one of the parameters from minimum to maximum value based on literature values, how much will this affect the model transfer efficiency? So on the figure here, the axis shows the transfer efficiency. Each row represents the change of one of the parameters, and the width of the bar gives you the resulting transfer efficiency from changing the parameter. So the very first one is the remineralization rate, and we've changed it from 0.03 to 0.3 per day based on values on the literature. In response to that, we had a change in transfer efficiency from 12 to 38%. Currently, the simple remineralization rate is really weakly constrained, and it causes a lot of uncertainty in the model prediction. In contrast, we've also changed, for instance, the temperature dependence from known values in the literature, and the transfer efficiency only varies by about 7% in response. Now, this is for today's transfer efficiency, and different parameters might be important for future changes. I personally believe that the temperature dependence will cause a change in transfer efficiency going into the future. But for the present day, this is what we found. Now I will move on to future predictions. So this is what the current CM6 models simulate for the particle export at 100 meter depth. This is the time series, so we have years here. The changes are shown in percent. Each line represents one model, and the black line is the multi-model. So currently, models show everything between almost no change and a decrease of about 20% in export, and there's one model that even shows a 30 to 40% decrease. I wanted to also remark on something that was discussed yesterday, to what extent the biological pump already responds to the human population, or how much it is responsible for taking up anthropogenic carbon. And so from the models until now, roughly, we see very little changes in the biological pump. I also wanted to show, though, if I show you the total values not normalized to 100%, then you can see that the differences between the models are actually significantly larger than the changes they predict. So I think from the modeling side, the uncertainties are actually just too big to be able to judge if the models interfered with anthropogenic carbon. If I had to guess, I would say rather no. Now, I also have these changes on the map. So this is the multi-model mean change until 2100. And you see that we expect decreases mostly in the North Atlantic, the upwelling regions, the tropical upwelling regions, and the subtropical gyre. regions. And there is a classic explanation of why this decrease in export occurs. As follows. So in the future warmer world, we will have a more stratified ocean. This stratification prevents mixing between the different layers. Because of that, there will be fewer nutrients mixed towards the surface ocean. Fewer nutrients means less food for phytoplankton to grow on, so there will be less primary production, and in the end, this will cause lower export. And I'm sure this is one of the main mechanisms that's going on in the CMIP6. We also know, though, from CMIP5 models that the exact way how this plays out can differ strongly between models. I've seen models where really primary production decreased, and the particle dynamics relative to primary production remained mostly constant. But in other models, there was almost no change in primary production. Instead, fewer particles were generated. And in yet other models, we had the exact same amount of particles, but the remineralization was faster, and therefore we ended up with lower. So I'm assuming part of the variation in projections that we see is because of the uncertainty in how the particle flux is implemented. So here is also the projection for the particle flux at depth. This is, again, efficiency. So the changes here are somewhat dampened compared to what we see at 100-meter depth. So models suggest something between almost no change and a slight decrease. But really, the changes aren't very strong. The strongest decrease is about 4%. And I do not know what is driving the changes here, but I can speculate a little. So I know for the NPI models, these are the two that show no change at all. I know that these models don't have a temperature dependence or oxygen dependence. All other models do. So I'm assuming that the temperature dependence of remineralization plays a role in the future decrease. I also know that the IPSL model is one of the very few models that has two different particle sizes. And at least in the CMIP5 projections, there was a shift towards smaller phytoplankton that creates smaller particles that sink slower. So this could also affect the changes in transfer efficiency. So I'm mostly speculating right now, so I move on. This is all that I wanted to show you regarding the flux of sinking particles. And I've been throwing a lot of facts at you, so I thought I'd give one quick summary slide before I move to the other two components. So in the default CMIP6 model, you will have one type of particle, it sinks with a constant speed, and it gets remineralized, maybe depending on temperature and oxygen. Very few models have mineral ballasting. Nothing else is implemented. At 100-meter depth, these models suggest something between 4 and 12 gigatons of carbon per year today, and are predicted to decrease between 0 and 40%. And at 1 kilometer depth, we have about 0.6 to 2.2 gigatons of carbon per year. And for the future, the transfer efficiency will either stay constant or slightly decrease. Okay, this is all I had about the sinking particles. I have two more slides on the dissolved carbon export and the zooplankton model, but this will be much quicker. So in terms of the dissolved carbon export, I feel there's a bit of a gap on the observational side. So all CMIP6 models I know have this implemented, but for the last 10 years or so, I can't find a multi-model comparison paper or any type of paper that actually looks at the dissolved carbon export and what changes we predict into the future. There's very little I can actually show you today. What I have is the surface DOM concentration in CMIP5 models. Again, this is not the export, that's just the surface concentration. But based on those figures and the differences that we see between models, I would expect that there are quite some differences in the dissolved organic matter export. Two models had a very nice and thorough model description, so I can share you the DOM export in that model. That was either 3.6 petagram carbon per year or about 1 petagram carbon, and it represented 36% or 14% of total export. I do not know anything about how the DOM export will change with future projections, and I also cannot give you an overview of how it's implemented in models. Also, time is running, so I will move on to the zooplankton part now. None of the CMIP6 models has zooplankton vertical migration implemented. I'm also not aware of any model study that attempted to do a future projection. I know 3 studies I know 3 studies that attempted to do a present-day estimate of how much carbon is transported by zooplankton migration. The one is a satellite-based model, then there is an Earth system model, and I also know the data assimilation model. And they all come up with an export of about 1 petagram carbon per year globally. And also, I know the figures are hard to compare because they have a different color bar, but the unit is the same. And to me, at least on first order, they show a similar pattern. So this is a reassuring first step from the modeling side. They have also all mentioned that it is, with the currently available data, impossible to robustly evaluate a model. And they've highlighted key uncertainties, which would be what fraction of the zooplankton migrates, to what depths do they go, and also how much they actually poo at depth. All I had prepared for today. Actually, there was a summary slide. Apparently not. Well.
All right, we're going to get started with our next 2 overview talks. So we'll have 2 more slightly longer talks, and then we'll go into lunch. So the first of which is going to be from Colleen Durkin from MBARI.
All right. Thanks for inviting me to talk today, and I'm really excited that Debbie and I are in the same group together because I think we have a shared mission to make everyone lovers of fecal pellets. So, although I'm not going to talk about fecal pellets that much today, I was asked to talk about I was asked to talk about how we measure the biological carbon pump, and I decided what I would really like to focus on is these potential ways that we can increase the time and space scales that we're observing the biological carbon pump. So for example, you've been watching this video showing examples of some of the ways that we do this now from the North Pacific export cruise. Yeah. All right. You can watch it more later. I'm going to go in front here. Okay. So measuring the biological carbon pump, it requires extensive resources, including personnel, ships, expensive instruments, and expensive laboratory analysis. And just, for example, this is a time-lapse of our sediment trap deployments from the Exports North Atlantic cruise. And I'm sure anyone who's done this work knows how much work it is. So, but these efforts are really important. All of that work forms the foundation of our understanding of the biological carbon pump, all the mechanisms involved. And so I'm highlighting here, I just decided as, illustration, I want to highlight all of my efforts in the last 10 years for measuring the biological pump. And I think that everyone else who studies the biological pump could make a similar map of their own. And imagine if we could put all of our maps on top of each other and how many dots we would have all over the world. But still, you'd have to look at it and say, well, they're still just snapshots in time and in space. And some of these dots are just a single dot. And so really the big question is, how do we scale up these mechanistic observations of the biological pump to improve the global scale quantification, monitoring, and modeling? And we just heard about a model from Dave and from Charlotte, and I'm showing you here this. I love this animation of the Darwin model, which is an ecosystem model of the surface ocean, but you can see the incredible biological detail that's being resolved. And this type of detailed biology is now starting to be incorporated into biogeochemical models like the Echo-Darwin model. And so the big question for people making observations is, how can we collect observations that are detailed at the scale that will improve these models? And when I talk about mechanistic observations, I'm talking about the quantity of sinking particles, the ecological source of those particles, and how they're transformed and attenuated with depth. And so I'm going to kind of split this overview talk about these methods in 3 parts. The first is optical and chemical sensors, imagers, and DNA. So let's start with the optical and chemical sensors. And the best example right now of really scaled up optical and chemical sensors that are capable of measuring the biological pump is the BGC Argo floats. This is a map of what's currently deployed today that has nitrate, oxygen, fluorescence, and backscatter sensors. And this is what those floats look like. You can see the photo there. This is Seth Bushinsky talked about this a little bit yesterday, but this is how they work. So, I get to use the stick. So they sink to 1,000 meters, and they float for typically 9 days, but that drift time can be varied. And then they descend to 2,000 meters and then do this profile to the surface. And it's really this repeated profiling that provides a lot of time-resolved data, and then it repeats the cycle again. So the way that it's used for monitoring the biological carbon pump is it can be used for chemical budgets, but I'm going to focus on the backscatter and fluorescence ways that it's measuring properties of particles. So I'll just show you one example here. This is a study by Nathan Briggs and his colleagues using BGC Argo floats. So the green lines here are those repeated vertical profiles of a float. And he used both the backscatter and chlorophyll and interpreted the anomalous spikes in that sensor as large particles. So you're seeing these large spikes of chlorophyll, which are interpreted to be large phytoplankton aggregates, and you can see that it can detect this sinking of large phytoplankton aggregates over time, and that's possible because it's sinking in this coherent pulse, right? And then the thing that was cool about this study is the smaller particle fluorescence appeared at depth about 2 days later. And so the outcome of this work was, in these cases, he estimated that about 50% of sinking carbon was attenuated by fragmentation during these pulse events. And so I'm just showing this as an example of one way that these sensors can be used to resolve some mechanisms of the biological carbon pump. And I want to highlight another optical sensor, the optical sediment trap. This is an instrument that has been developed a lot in recent years by Meg Estafa. And this is what it is right here. It's a transmissometer that is mounted vertically. Here it's on a neutrally buoyant sediment trap. And how this works is that when that float is drifting at depth, sinking particles land on the optical plate here, and it attenuates the light. So the beam goes between here, and that attenuation of light is a proxy for carbon flux. And Meg has made these measurements many times in many different ocean locations over the last 10 years, and has this really beautiful correlation. So the different colored dots are from different ocean locations, and there's this nice correlation between the POC flux and the beam attenuants. And so this is really powerful because it enables us to have this reliable proxy of POC flux without having to collect a physical sample. And I'll just show you an example of the type of resolution we can get with that. This is that float. This is that POC flux calculated from the optical sediment trap. And this is one drift over several weeks during the Exports North Atlantic cruise. The optical sediment trap proxy of POC flux, you can see there's this kind of moderate level of POC flux for a few weeks. And then it's able to resolve these short-term spikes in POC flux that happen. And that was that sudden export of the phytoplankton bloom that Dave talked about earlier. And so I'll also tell you to go see Meg's poster because she has some really exciting ideas about where to take this type of instrument. So you have an opportunity tomorrow morning to go talk to her. And so the takeaway here is that the OST, it detects a quantitative proxy of POC flux over time. So, I mean, that's certainly not a comprehensive review of optical and chemical sensors, but I hope I gave you a sense for that they can tell us how much carbon is sinking and the change over depth and time. So now I want to move on to imagers. And when I think about imagers, I think of them in two different categories. So there's imagers that detect all the suspended particles in the water column, so they don't distinguish between sinking and suspended. And then we also have imagers that specifically detect sinking particles. And typically these are cameras that are facing upward and collect a time series of images of particles that land on that camera, like here. So let's start with the suspended imagers. And I'm just going to highlight a few that are pretty widely used. There's shadow graph cameras, like so here I'm just showing an illustration of the ZOCAM, which is developed by Mark Oman at Scripps. It's very similar to the ISIS, which is another shadow graph imager that's commercially available. And the way these work is they are really powerful because they can focus through a large volume of water and keep those large particles in focus. So they're good at detecting large, rare things, which are really important. And then another type of imager, which has a slightly different type of optic system, but is very widely used and commercially available, is the underwater vision profiler. And this uses a sheet of light, and then when the particles go through that sheet, it detects the side scatter light. And both of these instruments, I'm showing them here mounted on AUVs and gliders, but they can be put on CTDs and to do profiles. They can be towed behind the ship. They can be put on moorings. So they're really versatile. And this is just an example of a map, like a three-dimensional spatial map of particle abundance that you get from the UVP. And this is what those images look like. So this is the UVP side scattered images, and this is the shadow graph cameras. And what's really exciting about these tools, and what I think makes them really powerful, is that they're seeing the particles in the water, but they're also seeing the animals and organisms that are co-located. And so we have this potential ability to both see the sinking detritus, or the detrital stuff, but also the animals that are producing those particles and transforming them as they sink. So we have this potential to really identify those mechanistic interactions. And I also want to point out that it's no small feat to deal with this image data after you collect it. And anyone who uses these instruments knows how hard that is. But there have been some recent advances, and I just want to point out all these papers to you in case you're interested. There's a nice review by Irison that reviews the current state of machine learning to do the classification of images here. Martin Schroeder has an exciting tool called MorphoCluster that could really help with the rapid annotation of these images. And then the study by Trendowska from 2021, and then my paper also from last year. They are good illustrations of the value of classifying these detrital particles, because it tells us the ecological source of those particles and also helps us model their carbon content. So you can take a picture or ask me later. So now we're going to move on to sinking particles. And I'm going to start by talking about imaging systems that directly measure sinking. So this is a study by Iverson and Lampett, and they mounted a sideways facing camera here on a neutrally buoyant platform. This is the Pellagra sediment trap, and did particle tracking to measure sinking speed in situ directly. And I'll just let the title of their paper explain what they found in this study. Was they couldn't find any relationship between the size of a particle and its sinking speed. And that might be surprising. It's not what theoretically should be true, but it's also been seen in the past. There's a few other particle tracking in situ studies that have been done. So Dierks and Asper also found no relationship between size and sinking speed. And there's a really beautiful work by Cindy Pilskin doing particle tracking using ROVs. And she did find a relationship, but it was highly variable. And so I just want to point this out, that this is a huge unknown, and I think, you know, Charlotte's talk highlighted how important just getting that number, sinking speed, is for models, and we still don't have a predictive understanding of what controls sinking speed. My own opinion is that we need to resolve what those particles are, because it's not all about size, it's also about what they are. And I predict that we'll see a lot of advancement in this area in the future, and if you don't know what to work on, I think you should work on this. I mean, it's the place where we have the potential biggest benefits, I think. Okay, so now I'm going to talk about other sinking imagers. So the upward-facing camera. So this is right here. I'll start with the SNOCAM. This is developed by Melissa Omond. It's an upward-facing camera. This is the imaging platform here. It's attached to a neutrally buoyant, the Lagrangian float designed by Eric DeSaro. This was deployed during the Exports North Atlantic cruise. And so how it works is it's floating there, and then particles settle on that imaging screen, and it just takes time-lapse images of sinking particles. So you can see this is what the images look like, and this is after going through image processing, so we can detect the particles, right, and quantify them. And not only can we quantify the area flux of particles, but because they're images, we can also identify what they are. So in this case, we could distinguish between aggregates, fecal pellets, and even some organisms. And so, this is the same graph I showed you earlier with the optical sediment trap. So what you can't see here is that optical sediment trap was actually, you can't see it on the Lagrangian float, but there was an optical sediment trap. And so that data I showed you earlier is actually the time series of the optical sediment trap on the Lagrangian float. That's when we saw this time-resolved pulse of sinking particles through the OST proxy. Now, the SNOCAM was right next to it, and now this is the same exact graph, and I've overlaid the gray line here is the area flux detected by that camera, and it really nicely corresponds with the OST. So, great sanity check. In addition to having the area flux, you know, we also have images, so we know what those are, and that's what I'm showing you down here. These are what those particles actually were based on area, and there was a consistent sinking of these small particles that are less than 500 microns that are, for this particular camera resolution, we couldn't identify what they were, but there's a little bit of sinking from fecal pellets, but you can see that most of the flux in these kind of pulses was from those phytodetritus aggregates. And I want to highlight a few other imaging, upward-facing camera, flux cameras. Melissa has adapted that SNOCAM design into a standalone platform, so it's a standalone float called the Minion, and this is, again, an example of the time-lapse images and the image processing from that. And then Jim Bishop has also designed the Carbon Flux Explorer, which is an upward-facing camera on this sediment trap that's attached to a neutrally buoyant float. And so you can see what those images look like, too. And I would point you to his student, Hannah Bourne, has a couple recent papers that are really nicely explain the use of that. And the last imaging sediment trap I want to talk to you about is actually designed for the deep ocean, so 4,000 meters. This was designed by Ken Smith's group at MBARI, and it has been deployed at the Deep Ocean Station M time series for long periods of time. So the sedimentation event sensor is a modified McLean sediment trap, which you might recognize, but instead of having collection bottles, it actually has an imaging plate, and it can take images however frequently you want. So they often have it set for a 2-hour resolution at 4,000 meters deep, which is incredible. And this is, so again, an example of the images, the time-lapse images, and, you know, you can put it through a processing program to quantify that. And I'm going to show an example of the type of data you can get out of that, which to me is just, it's amazing. I mean, you're at the deep ocean, and we can monitor daily average carbon fluxes in this really remote and hard to sample depth. And so this is just one 9-month period where it was deployed, and so you can see the modeled carbon flux based on the images. And what's cool is there are these, single-day episodic fluxes which would never be detected in a 10-day or 14-day integrated flux collection, which is how they normally are done. And we know that they were because of these random arrivals of salt fecal pellets. And then in the spring, we see this big pulse of flux, and we know that it was because of salt fecal pellets. So I just want to illustrate to you the power of these biologically resolved observations of flux. Imagers can tell us how much carbon is sinking, the change over time and depth, the ecological source, and the animals that, and in some cases, some imagers, the animals that transform and produce those particles. Finally, very briefly, I'm going to talk about DNA. And so, DNA is very powerful because it's not going to be good at actually quantifying carbon. It's not a way to measure carbon, but it can tell you the ecological source. So more like a forensic tool to make those linkages. And there are a lot of really great work, especially I'd say in the last 5 years. There's been a lot of papers published on this topic. But because I don't have that much time, I'm just going to talk about my work. So I'm going to show you a recent study that we published from the California coast. The data I'm going to show you is from there. And this was from a research cruise led by Ivana Cetinich and many other collaborators. So we deployed sediment traps. And this is, the collection tube. And some of them collected bulk sinking particles for DNA sequencing. And other ones had a gel layer at the bottom. And this is what a gel layer looks like. This is a really great way to collect sinking particles to get good images of them, distinct particles. But the other thing that's nice about it, this video is not going to work. Is we can isolate, we can use it to isolate individual particles with a pipette and then extract the DNA from individual particles and link those to particle types, right? And so this is what we found in that California coast location. These are the 5 phytoplankton functional groups we detected in the surface mixed layer. And these big boxes here, for example, the big box under diatoms represents all the taxa that were detected in the surface mixed layer and their relative abundance. So these are just genetically distinct sequences that are assigned to diatoms. So for example, we can actually put species names or organismal names on each of those boxes based on databases, what they're most closely related to. And this is like the relative abundance of this organism in the surface based on sequences, right? But we also sequenced what was in the sediment trap, and so now I'm showing you here which of those taxa we actually detected in sinking particles, so we can start to link who was in the surface, but who was actually sinking. So again, these aren't really carbon units, so we tried to push this farther, like how could we actually use DNA in a really quantitative way to advance biological pump models? So this is the same exact figure, but now I've scaled these to the relative carbon biomass of each of those functional groups in the surface mixed layer. And we did this using HPLC pigments. And the exciting thing about using HPLC pigments is that's kind of relatable to what we might detect with an ocean color satellite. So we can think about how you might be able to scale these type of measurements. And of course, I'm sure somebody's going to ask me a question. There's a lot of caveats here, but we're trying to think about how, just trying to move things forward, right? But so now that because we measured the sinking flux, well, this graph didn't work, but there's supposed to be flow. Okay, so we can trace the amount of carbon that goes from each of these boxes down into different particle types and in a quantitative way. And if you want to see this actual figure, then you should You should read my paper. But there's a beautiful, I mean, basically we can put quantitative arrows from each of these into, in this case, we were able to distinguish between the fraction that goes into small particles. I think in that case it was less than 300 microns, and large particles. And I think as we get more data, like from the NASA exports, I just sent off 1,000 particles to be sequenced last week, so we're going to have a lot more data. I think we'll be able to track it into very specific particles as well. Oh, right, and because we did the imaging of the particles, we know how much carbon was transported by each of those particle types. So like those numbers on there are carbon numbers. So DNA has the can tell us the ecological source, the animals that transform and produce those particles, and can identify the ecological links, especially in that case it was surface to sinking. And that is what I have for you. Thanks for your attention, and I hope you have questions.
Wonderful stuff. Thank you. I really enjoyed your talk. A question about your DNA work. I should say who I am. I'm Tammy Richardson from the University of South Carolina. Your probes, are they just eukaryotic probes?
Oh, yeah, I should have said.
I was looking for the cyanobacteria. You know that.
That was just 18S, so that's eukaryotic, right? So that's why there was no cyanobacteria in there. For exports, Alison Santoro did the 18S sequencing, so we'll get all the chloroplast and cyanobacterial DNA from that. Yeah.
Hi, this is Charlie Stock from NOAA GFDL. Your deep example highlighted the value of long deployments, and I was just wondering if you could comment on the potential for longer deployments of some of the more pelagic systems. Can we have a fleet of BGC Argo export measurements out there?
Yeah, I think you should go to Meg's poster.
Okay.
Yeah, I think that's the goal, whether or not it's integrated with BGC Argo or it's a separate thing. I mean, like the minions, my understanding is, power use is a huge thing depending on how long you want it to be out there. The amazing thing about that deep ocean trap is it's really low power. And so you can put it out for long periods of time. And one thing that we've been thinking about with that particular trap is the potential for it to just be like, it's the low power sensor that's out there and kind of monitoring. And then when something cool happens, it could signal to other high powered instruments and tell them to turn on and say, oh, now, don't waste your battery when things are boring, but now we need your measurements, so turn on.
Thank you.
Yeah.
Hi, I'm Megan Sullivan from UC Irvine, and I had a question on how do you account for the variations in stoichiometry when getting from the sinking flux to a carbon unit? So I saw you accounted for like what the different organisms are, but even within an organism, we know that it varies, and then also from the surface to sinking, they can, the carbon can degrade at different rates from the nitrogen and phosphorus, and I was just wondering.
Oh, there's no
account.
So it depends on if you're talking about the particle carbon, trying to model that. That is done. So each particle type that seems like it made sense to put in a different category has a different equation that converts volume to carbon, and so. Yeah, I could imagine a cool model where you make that more adaptive and the maybe, the zooplankton, they're kind of not eating very well at this time of year, so you might want to reduce the carbon. You maybe want to alter that equation so the carbon content of their fecal pellets goes down or something like that. But with the phytoplankton, that modeling of the phytoplankton carbon was based on an HPLC model, which of course isn't perfect. And there's other ways to get at it too, right? You could have like IFCBs out there and get imaging or cell counts even, right? But it doesn't take that into account, but I could see that as being a cool future incorporation into it. Yeah.
Hi, Hillary Pilevsky from Boston College. Colleen, that was an awesome talk. And it's really amazing seeing like all of the additional information we're getting about the particle types and the connections between the surface and the deep ocean, particularly from the imaging and the DNA work. And so I'm thinking about the connection back to Charlotte's talk and like, okay, as in our observations, we're just proliferating the kind of information we can collect about all the diversity within the particles. And then like, is there a way you see as the path forward to synthesize this into something to say like, what of this information needs to be passed on to the modelers? Like, are there more particle types that are important to the models, but not in the like, okay, now we have the whole kitchen sink and we have to throw the whole thing in?
Yeah, I think I'm just thinking about some of the kind of synthesis discussions that have been happening and trying to come out of exports, right? And I feel like we're starting, I could imagine being like, okay, you can't put 10,000 particle types in there, but what about, you know, salp fecal pellets, or like things that think really fast, and what about just dividing it into things that are like, think fast and don't attenuate and are big, or things that get repackaged with depth, or, you know, maybe like 3 different pathways like that have different effect on the attenuation. Because I definitely, when I saw the way that attenuation or transfer efficiency was modeled, I thought that was very depressing. Like a single size particle with a single thinking speed, that's like the opposite of all the work that I've focused on in the last few years. So I thought, you know, it seems like very simple, but I get that it's really complicated and you can't put everything in, and you don't need to necessarily put everything in, but it's totally unrealistic, right? So there might be a way to have a compromise.
Excellent. And I'll add, I think it gives you a wonderful opportunity to mechanistically link the surface to the deep, and also quantify how important are episodic events, and if you average across them, does it all come out in the wash?
Yeah. Was there a question in there?
No. I was just adding to your awesome data.
No.
And we'll hopefully can pick up on these new methods this afternoon in the breakout groups, and then synthesis to follow.
Yeah, and all the talks, Debbie and all the other talks, it's going to build on examples of that.
Thank you so much.
All right, so we're going to move into our last talk for overviews this morning with Debbie Steinberg from VIMS.
All right, well, thank you for having me. It's great to be back here. And I am going to talk to you about the biology of the biological pump and episodic and ephemeral nature of carbon export. And I just want to acknowledge some of my collaborators on the export project that worked with me on some of the SALP paper stuff I'm going to show you shortly. And yes, that is not Helvetica font in my name, you might notice. That is my name written in fecal pellet font. And yeah, kind of special, I think. So I'd also like to acknowledge the Exports Project and funding by NASA for some of the things that I'm going to talk about. I've been thinking about this question for a long time about the episodic, ephemeral nature of particle export. And as far back as I can remember, I think the first time was when I was in graduate school at UC Santa Cruz, where for my dissertation, I was looking at zooplankton associated with marine snow. And I did a lot of blue water scuba diving for that project. And one day, I was sampling in Monterey Bay, California, which is during the spring, and there's upwelling, and the water's really soupy and green. And I had a series of sampling days, and I went out one day, and we were diving, and I couldn't see a thing. It was pea soup. There were no aggregates, but there was definitely a bloom going on. So I went back the next day. We went diving, and the water was beautiful blue, and then there were these giant diatom aggregates everywhere. So it was blue, crystal clear blue, but with these giant aggregates. And I was witnessing aggregation, right? And so I collected in my little syringes and brought it back and looked at it, and you can see some of what the particles look like here. They're full of diatoms. And then I went back the next day to get more, and it was totally gone. The water was blue, and the aggregates were gone, and I didn't get any more samples, which was a bit of a bummer. But I said, wow, that happened fast, and wasn't I lucky to catch that? So that's my first experience, I think, with this issue. And so what I want to talk about today is some more examples of that, just to prime the pump, so to speak, for our conversations later, and talk about deep ocean carbon flux, zooplankton-mediated export, talk about gelatinous zooplankton blooms and seasonal vertical migration, and we'll talk a bit about phytoplankton blooms and give you a few examples of some episodic export from diatom resting spores and viral infection, and then I'll just leave you with a few parting thoughts. So first, Colleen mentioned this and introduced this time series, which was great, the Station M long-term time series. And what this is showing is this episodic organic carbon export to abyssal depths in the Northeast Pacific. And this is a 29-year time series in the California current upwelling system. And at the very top there is, shows you the temperature time series. And the solid lines across is the long-term mean, and then the dashed line are two standard deviations. And so what this study was looking at was these episodic flux events, and anything above those dashed lines is something they're calling a episodic extreme event. So first of all, this is the temperature time series here. You can see some of those El Niños, the warm water. This is estimated POC flux from the euphotic zone from satellites, and the chlorophyll is in green there, and the black is the POC flux estimated from the satellites in the top in the euphotic zone. And then here we have the sediment trap POC flux at 3,400 meters, that deep trap that Colleen showed you. And what they found is that 19% of the annual POC flux and this very deep trap arrived during these high magnitude flux events, and you can see them here. And they seem to increase with time, because 43% of the annual POC flux in that deep trap arrived there in the later part of the time series. And maybe you can see a little bit better here. This is the percent of the annual POC flux in the traps, and this is and those episodic events are in these dark colors here. So you can see that seems to have increased over time at this site. And so they make the point that not all flux events from the euphotic zone that were estimated from chlorophyll and from satellites were translated into abyssal POC flux. And so that flux model just from the satellite data alone wasn't good enough to predict the development and also the fate of these high flux events, because we can also see this material getting onto the abyssal seafloor as well. Colleen just showed you a version of this, and I just wanted to show you this to say that, you know, it's during some of these episodic events, sometimes there's just one kind of a particle that is sinking down, and in this case, as you just saw, these were salt fecal pellets. And so this is a picture of a salt fecal pellet. And so the salt fecal pellets were really important. These episodic events were very important to deep POC flux. And during that 8-month sampling period in this study from that optical sediment trap, about 45% of the total POC flux at 3,900 meters was coming down in these salt fecal pellets. And so, yeah, so that's what we're seeing there. And then that was also, those pellets were also a source of chlorophyll peaks and phytodetritus at depth. And this was making it all the way down to the bottom of the ocean. Another example of episodic flux is hurricanes. And so we're going to move now to the Sargasso Sea off of Bermuda. And this is example I'm showing here is when Hurricane Nicole passed over Bermuda in 2016. And when this is, you see the, this is the velocity and the surface waters, you can see the hurricane. This is the track of the hurricane and the sea surface temperature cooling, mixing up this colder water from deeper. You can see the chlorophyll increasing. This is what the sediment trap time series trap looks like, the ocean flux program. And this is the mixed layer deepening as the hurricane is passing over. And what they found when looking at this material in the traps is that there is this pulse of very highly labile organic matter, lipids, and from zooplankton and phytoplankton. And this right here is when the hurricane arrived, and you can see the increase here in lipids and phytodetritus. And then that's in the 1,500 meter trap. And then we come over here to the 3,200 meter trap and see that material arrive at depth after the hurricane passes. So these are these pulses of this really organic, rich material that feeds the deep sea. The next thing I want to talk about is zooplankton and the biological carbon pump. And this is the zooplankton ecologist-centric view of the biological carbon pump. And Dave and others have already explained the different mechanisms here. I'll just reiterate that there's sort of 3 different ways that zooplankton mediate export out of the surface waters by feeding in the surface waters and producing sinking fecal pellets, and by diel vertical migration, which is feeding in the surface waters at night on phytoplankton that have been incorporating carbon, and then swimming back down to their daytime residence depths in the mesopelagic zone where they metabolize that material, and we call that an active transport of carbon to depth. Another way is thinking of carcasses, and in particular, gelatinous zooplankton carcasses, and that is another way that some of this material gets down to depth. And I'm going to highlight the role of gelatinous zooplankton here when in the context of episodic flux, because they can often play an outsized role in the biological carbon pump, and a lot of that has to do with their life history. There's several kinds of gelatinous zooplankton that form these episodic blooms because they have both a sexual and an asexual stage to their reproduction. So they can form, their populations can rapidly increase. And some kinds, like salps, have very high filtration rates on a high or broad size spectrum range of particles. They produce large, fast-sinking fecal pellets and undergo extensive diel vertical migration. So if they're migrating to great depths, that's getting this carbon down to sequestration depths, which Dave was talking about, and Charlotte and others. And the sinking of their carcasses after the bloom demise is very important too. To show you an example of that, these are some photos from the seafloor of different kinds of gelatinous zooplankton around the world that people have captured on cameras. Over here, we've got pyrosomes, which are colonial tunicates off of West Africa. And these are them here. They just kind of collect on the seafloor. These are some medusae in the Arabian Sea, 1,400 meters. There's just a big pile of jellyfish at the bottom of the sea there. This is one of my favorites. This is Nepolemia, those giant jellyfish off of Japan that bloom sometimes. Note that the diameter of the bell of this jellyfish is almost a meter, okay? Then we've got, this is a salp bloom that fell in the Tasman Sea, and that's a crab feeding on it, and here we have a shrimp feeding on a pyrosome. So this stuff is making it down to the deep ocean. The example that I'm going to spend a few minutes on is from our exports project at Station P in the Northeast Pacific. And we encountered a bloom of Salpa aspera. And this is Salpa aspera swimming around in a beaker. And salps, if you're not familiar with them, are gelatinous zooplankton. They're kind of like a barrel, and they have muscle bands around them. And they move through the water pulsing, and as they're moving through the water, they're filtering particles out of the water on an internal mucus feeding mesh that can capture bacteria-sized particles on up to large diatoms. This is the salp's gut. It's making a fecal pellet right there. Woo! Pooping in action. And we encountered this bloom, and the night waters were just full of salps because they were vertically migrating. And so there's a graduated cylinder full of salps. This is a picture of their fecal pellets here. So, all right, that's what I want to say there. So this shows you the diel-vertical migration of the salp. And what this is, these are data from the mock nest, and Dave showed you some of this for the North Atlantic. This is from the North Pacific. We deploy the nets, and we get these discrete depth net tows, and this is the day and the night tow, and these are the different size fractions of zooplankton, and then the yellow are the salps. And what you see are the salps moving from mesopelagic depths during the day up into the surface waters at night. And the other zooplankton are doing that too, the crustaceans as well. And you can see it here, and then they are doing it there. So these they're feeding in the surface waters at night, and then they go back down to depth where they metabolize, and so they're actively transporting carbon from surface waters. And they're producing fecal pellets. And you've heard already a bit about salp fecal pellets, and I wanted to just give you a sense of how big they are compared to other zooplankton fecal pellets and why it kind of matters what zooplankton are out there. So that first graph there from one of Karen Samyeshkin's papers shows you the comparison of fecal pellet production by salps in blue versus some different crustacean zooplankton, amphipods, copepods, and euphalsids. And they feed at very high filtration rates. But it's not just the rate of feeding by salps, it's also just the size of their fecal pellets. And it struck me that these little chocolate croissants that we this looks a lot like a salp fecal pellet, okay? And this is a Tic Tac. Okay, so but we're going to compare the size of fecal pellets by different zooplankton. This is about the right, that's about right, okay? We tried to find green Tic Tacs, but for phytoplankton, but anyway. So this is just to give you a sense of why it matters, right? And I'll pass these around because I don't need, maybe you could pass them around, I don't need them anymore. Okay. All right, so they're big, they sink fast, like 1,000 meters a day compared to maybe 100 meters a day for a really big copepod fecal pellet. And they also are making up a large part of the fecal pellets that's produced in the surface water. So this shows you day versus night proportion of fecal pellet carbon produced. And the blue, again, are the salps. So sometimes they dominate the fecal pellet production in surface waters. We saw these fecal pellets throughout the water column because luckily we had an underwater, or it should be vision profiler here. This is a picture of a salt fecal pellet from the UVP on the cruise. And you see these spikes here. The bars here are integrated numbers. And notice here this spike at nighttime in surface water. So we saw these fecal pellets throughout the water column, and they ended up in the sediment trap. So here are sediment trap profiles. The blue is the proportion of sinking POC that are salt fecal pellets. We put it all together to look at the importance of salts in the biological carbon pump here. And what this shows is a schematic. This is the total POC export from the sediment traps, and that actually does include salt fecal pellets. And then we can separately estimate or measure the salt fecal pellet export from Colleen's gel traps and the DVM flux from knowing the change in the salt biomass in the surface during versus day versus night, and their metabolic rates, which we measured, and also the sinking carcasses. And we put all that, so this is the mean and then the maximum carbon export across 100 meters by those different salt-mediated export processes. And what we found is that salt-mediated export increased the biological pump efficiency. So the EZ ratio, so that's the ratio of POC flux at the base of the episodic zone to net primary production, increased by 1.5 times with the addition of salt. And then the T100, so this is transfer efficiency, but we're looking at it across 100 meters, and that's the ratio of POC flux 100 meters below the episodic zone to the POC flux at the base of the episodic zone, and that increased to about 2.5 times. So salts are increasing the export and transport efficiency of the biological carbon pump. And they occur, it's not just our cruise, and we got really lucky. We know from the time series at Station P that salts do occur there. So we know they're occurring mostly in the summer, and this is the time series showing salt blooms. And they're episodic, right? And they're not there all the time, but we know that they do occur at other times. And other places too. So this is from the Palmer Antarctica Long-Term Ecological Research Program. And we have years where we have these big salt blooms. It's usually much warmer years. And those are salt filling that plankton net. And it is not fun to process that net. So there's just another example. We find these are all over. All right, I just want to mention a couple other things. Seasonal episodic active transport. So we've talked about diel vertical migration, but there's also seasonal migrations of zooplankton where they're feeding in the surface waters during the springtime. This is for Calanus and Marchicus in the North Atlantic, and they build up these lipids during the springtime, and then in the summer, they go down to their hibernation depths in the deep ocean, and they're carrying that carbon with them. So they're building these carbon-rich lipids for hibernation, but it doesn't strip the surface waters of nitrogen, and the carbon is preferentially sequestered in the deep ocean. And this lipid pump, we call it, is actually comparable to the deep POC flux in the North Atlantic. Same thing happens in the subarctic North Pacific with Neocalanus, happens in the Southern Ocean as well. And this is a modeling study using the continuous plankton recorder survey for copepod abundance of these diapause copepods, and a fitness optimization model. So it's a trait-based model for looking at changes in the seasonal diapause flux. And what this shows here, this is the average over the time series, and then this is the change in that diapause flux per decade. And what they found in in this modeling study is that this relocation of the dominant hibernating species due to climate warming causes this northwest shift in export. All right, just a few minutes left, so I'm going to just mention some of the phytoplankton episodic export. And a really interesting one is export of diatom resting spores. Here we have a study from Tatiana showing that these diatom resting spores of just a single species of diatoms dominated sediment trap material in the North Atlantic and comprised 9 to 64% of the sinking POC. And she estimated that probably came down within a 10-day period. And these spores have very high carbon content. They're resistant to grazing. And this is another study from the Southern Ocean, same thing. The gray here is the sediment trap total POC flux. And then the blue bars here are from 2 different species of diatom resting spores. And again, they comprise about 60% of the annual POC flux in just diatom spores from a couple of species. All right, I'm gonna just show you, this is the last one, viral infection and carbon export. I think, at least I did, tend to think of viruses as what is called a shunt for organic carbon. So viruses attack phytoplankton and bacteria, they burst the cells, you get DOM, it's recycled in surface waters, so there's not a lot of export. But in fact, there's a lot of more recent studies showing that it's more of a shuttle. So there's mechanisms by which the viruses cause TEP production, transparent exopolymer production, these sticky polysaccharides that cause aggregation. And this is a study and leading to export, so it's more of a shuttle. And this is satellite imagery of a Meliana huxleyi bloom, a coccolithophorid bloom in the North Atlantic. And using these very clever lipid markers and also DNA, we're able to detect the stage of infection of the coccolithophorid bloom by viruses. So early infection, late, and post-infection. And using optically derived flux measures found that the maximal infection the maximum flux, rather, was during this early stages of the infection, and then the flux decreased in the very late stage. And you can see some of these coccolithophore aggregates here. And it's they attribute it to this TEP production. All right, so just a summary and parting thoughts. These episodic events are significant in the ocean, carbon budget, and we need to capture this variability using data and models to fill in gaps, new technology. And it's very closely tied to changes in plankton community structure and species life histories, like gelatinous zooplankton, asexual phase of reproduction, copepod, diapause, diatom resting spores, and there's lots more other examples I didn't have time to talk about. I'd like us to consider the role of stoichiometry in export. Okay, sometimes there's very carbon-rich, these carbon-rich zooplankton, for example, that are going into diapause. And that climate change could alter the intensity and frequency of episodic events, increasing or decreasing the strength of the biological carbon pump. So thank you very much for your attention. Happy to answer questions. Yes, Bethany. Oh, Mike, yes.
Thank you for that. Feeding graphic. I'm Bethany Jenkins from the University of Rhode Island. I'm super curious, Debbie, to know more about the feeding strategies itself, because they seem to be super successful in these nutrient-limited oceans, like these chronically nutrient-limited oceans. And so as we think about increases in stratification, the fact that, what can you tell me about what they eat?
Yeah, so Salps actually prefer to be in lower chlorophyll waters because their feeding mesh can get clogged. So we tend to find them in more open water, lower chlorophyll, warmer waters. And so they are, for example, in our work in the Southern Ocean, we think that, they're going to be a climate change winner because as it's warming, we're getting more salps, and they can feed on smaller particles because of that very fine mesh. So their feeding ecology is such that they can feed on much smaller particles than can, say, copepods or krill and things like that. And that's going to change the biological carbon pump. And you should see Maya Thomas's poster for that. Talks about what effect that might have, on the biological carbon pump.
Katsumi Matsumoto from NSF. I have a couple of quick questions. If I read one of your figures correctly, the one that you showed flux after a storm, I think there was more flux at 3,900 meters than at 1,500 meters. So I wondered if you could comment on that. And then the second is this stoichiometry of export. So fecal pellets are important to understand, but zooplankton tend to be more homeostatic than phytoplankton. And so I wonder what the CNP ratio of fecal pellets are, whether they reflect like nutritional imbalance.
Yeah, so taking your second question first. So the CN ratio of a typical copepod might be 4 or 5, and the fecal pellets CN ratio is usually a little higher. The thing is that some of these, for these migrate, what I'm getting at with the stoichiometry is this preferential export of carbon. So those diapods and copepods have their C/N, we measured in a different study, sometimes like 19. And also there are, I think with phytoplankton too, depending on whether there's nitrogen fixation happening in the upper water column, that some of those particles sinking out are not at Redfield C/N ratio either. And I think that really matters for the biological carbon pump. So that's just one example for zooplankton and for phytoplankton. And then your first question, I have to go back and look at, but I think, I have to look and see why it's higher. But I will say that it's not unusual for us to sometimes see higher flux in a deeper trap than a shallow one. And sometimes that's because it's for a lot of different reasons. It could be advection of particles from outside the area, or other processes that are happening in mid-water, repackaging of particles in the mid-water into larger, faster sinking fecal pellets, for example, which could increase flux in a deeper trap. But yeah, I'll have to take a look.
For our first talk this afternoon, we're going to have Martina Doblin speak to our group here. So Martina, without further ado.
Thanks, Emily. I hope online you can hear me. It's fantastic to be here. It's my first post-COVID meeting, and wonderful to be with you in person. And for those joining remotely, if you're struggling with jet lag or you've missed out on that experience, good for you, because it is one of the challenges of meeting, obviously, internationally. It's my pleasure to tell you this quirky, interesting story of our research recently completed. But I wanted to say, firstly, that this is a collaboration between the University of Technology, Sydney, the University of Edinburgh, the University of Tasmania, and the Sydney Institute of Marine Science. And I did want to start with some thanks. The postdocs that did a lot of the lab work, Michaela Larson is a superstar. Any PI would want to hire her. She's amazing and really was the driver behind a lot of the observations. And I don't know how many hours of microscopy she did. This work was recently published in Nature Comms, and so please, I invite you to look at it again and go to all the videos and so forth. The work was funded by the Australian Research Council, and we really drew on a large body of observations from several ocean observing programs. So highlighting again that this is very important. And I really also wanted to thank the inspiration of the work of mixotrophy researchers, and obviously there are more in this crowd. Mixotrophy is the future. So thank you to the OCB meeting organisers and particularly to the chairs of this session. And it's really wanted to thank you for listening. A really lovely tradition of Indigenous Australians is to bring a branch of a native eucalyptus to a country, a part of the country that you're visiting. And so this is the eucalyptus tree in my backyard. All right, so this is a really lovely animation of Prorecentrum balticum, the mixotroph in question. It's swimming behaviour that really led us to really observe the sort of curiosities of this organism. So it would spend hours spinning or shaping what we've called a mucosphere. So it's exuded an exopolysaccharide. We know this from its positive staining with pumarcine blue and alcium blue. And it kind of forms this 100 micron diameter, almost like bubble-like feeding trap. And it only can do this once it's been able to photosynthesize for some time. And it can only survive when it is able to photosynthesize. It cannot sustain itself by eating. And so it's an obligate phototroph, facultative phagoheterotroph, and a constitutive mixotroph in their definition. So this exudation of TEP was a very interesting process, and it allowed us to continue to our observation of what happens next. So intriguingly, the mucosphere also contains chemicals that chemotactically attract microbial prey. And so this is not a passive process. And so already this DOC has become POC with no further physical aggregation mechanisms. So it's a really kind of new ecological process for particle formation, and not just the size of the phytoplankton cell, but obviously something much bigger, so it's about 100 microns in diameter. And it's obviously now ballasted with lots of other microbes in it. But the host, the Prorecentrum, can then select a prey item from this open buffet that it's surrounded itself with, and can basically pull out its peduncle and suck up, the cell of choice. As you can see, when you use the fishing rod, you can see this algal cell being consumed. So after it's consumed that cell, it will discard the mucosphere. So it can certainly get around behind itself, but it abandons it. And so in that process, it's negatively buoyant, and it started to get us really intrigued about what the contribution might be to carbon fluxes. Before I go there, we wanted to really take this lab observation and really ask the question, all right, so how prevalent might this organism be? How abundant is it in the ocean, and is this a significant kind of process? And we were able to use observational records of the plankton to observe this cell's abundance. And you can see around this sort of continental shelf observing program, there are 9 stations, and this organism is found at all of them, obviously episodic in its abundance, but with lowest mean cell abundance of some 19 cells per litre, but a maximum bloom type cell abundance of over 3,000. So reasonably abundant for a eukaryote of that size. And this happened in review, I must confess. We were then really asked, well, continental shelf organism, how prevalent is it in the open ocean? When we went back through JGOS and CPR records, we did see evidence of proricentrum-like cells. Remember, these cells are relatively small, not always taxonomically distinguishable from other cells, but nevertheless, we saw records of this proricentrum genus, and we think highly related species to the Balticum species that we had discovered. When we looked at gene sequences, though, we found an exact match in the Tara Oceans data set, and this allowed us to really explore the global distribution of this organism. And so this is a visual summary of its biogeographic distribution in the ocean, and you can see that, yeah, it reaches quite significant cell abundances, 6,000 to 8,000 cells per litre, and largely in the 5 to 20 micron fraction. Yeah. And notably in part of the water column that sort of spans probably below the mixed layer as well. So this became even more intriguing to us. And so when we thought about, well, how much carbon is this organism producing in its mucospheres, and what is it capturing chemotactically, and what does that really contribute to the biological carbon pump, or what is its potential? Obviously, there's a lot of caveats on how rapidly these things might sink, etc. What we found with direct measurements of the mucospheres, we created them under conditions of, so they were in azenic culture, they had had a period of light, they were able to make mucospheres, we were able to isolate those mucospheres and do a direct carbon analysis. And when we scale those estimates up, we find that they're reasonably significant for a single organism to make a contribution that represents about 0.1 to 0.7% of the estimated total carbon export from the episodic zone. So it was a reasonably significant number, and something that kind of represents a potential new pathway for making particles. So the mucospheres are not presently considered a major particle formation mechanism, and the fact that they're negatively buoyant and already contain 100 microbes together into this DOC bubble means that they could in fact sink quite quickly. So super intriguing to see some of the talks this morning about, well, could we in fact start to link this organism's surface waters to the particulate flux through different depths in different parts of the ocean. So here I wanted to just mention that the production of mucus isn't a kind of just a unique thing for this organism. It's actually reasonably common amongst dinoflagellates, at least, in this sort of mechanism. And it's not just the exudation of DOC, it's used to capture prey. And this is, I guess, an important distinction rather than just the DOC floating loosely in the ocean. This is actually capturing particulate matter as soon as it's released. So there is an ecological significance to it. I do want to say that there's some open questions, and as much as I've enjoyed listening to all the talks, I always think it's always a great opportunity to link with other people and say that there's things that we still want to find out. So we want to know that, we know that mucosphere production happens on a daily basis, and that it may not be episodic, but it certainly would be related to the relative abundance of this organism. But we really want to understand how might this behaviour change under future ocean. And really, we'd started with that question, and we didn't quite answer it. So very intrigued to see what the mixotroph groups do. We also would be interested in comparing this organism with other mixotrophs, whether there are different strategies that vary from continental shelf waters to the open ocean. And I suppose here I wanted to sort of make the case. We've talked a lot about models driving observations. Maybe this is something we want to talk about in the breakout groups, about observations showing us that perhaps models need to respond to the new insights that we're finding in the ocean, and so where the biology might be driving some of the model development, or at least testing to see whether mechanisms might be really important. So I will leave it there. I want to thank you for your attention, and it's 0 minutes. Done. Thank you.
We have time for one question as we transition to Heather McNair, who's going to be presenting virtually. Do I have any questions? Me?
You mentioned on your last slide P limitation, and I was going to ask you, is there any link of this mucilage production to nutrient limitation? So is that a yes for P?
Yes.
But what about, and what about N?
Not for nitrogen, but we do see increased growth when prey consumption when the dinoflagellate is also in medium without vitamins. Yeah, so it's a very intriguing story.
One quick online question from Laura Holland. What are the chemotactic signals used?
So we were able to establish through a chemotaxis assay that it was a bulk extract from the mucospheres into an organic solvent. And so it was basically the active movement of bacteria towards this mixed extract in a device that enabled us to test chemotactic behaviour. So I couldn't tell you which specific compound it was.
All right, and next we will have Heather McNair joining us virtually from URI.
Hello, thank you all, and thanks for the opportunity to do this remotely. And it's so nice to actually have an audience in front of me. All the back of your heads look very great today. So I can still feel the energy in the room, even though I'm talking to you remotely today. And I'm excited to share with you some of the work from the exports campaigns where we're linking food web mechanisms to export pathways and from the North Pacific and North Atlantic exports campaigns. So as Dave talked about earlier, the goal of exports is to get to a predictive understanding of the fate and export of net primary production in the ocean. And so I've put together this schematic here that kind of helps me understand what we're thinking about in terms of linking these surface processes to these export pathways. And so we have our net primary producers here, and these arrows representing flux of carbon. And so our net primary production can be assimilated into the larger food web, with portions of it then being exported via these food web processes. So sinking of fecal pellets or diel vertical migration, things that Deb and Colleen talked about earlier this session. Or we have this net primary production that can be exported directly via physical mixing, sinking of cells, or sinking of aggregates. And so there's been a ton of work done for the exports program on these food web processes, but I want to narrow in here on this first link in the food web, this role of microzooplankton grazing and how that's allowing us to kind of push forward and get to this predictive understanding of how carbon is moving through the system. And so microzooplankton grazing has become kind of a special piece of the puzzle that enhances our understanding because it shows us how much carbon is being assimilated into this larger food web, but it also tells us the amount of primary production that remains unassimilated into the food web that can then be directly exported via these other export pathways. And so the cruise design of exports really gave us this unique opportunity to take bottle incubations where we're studying a very specific process and compare that to what we're seeing with in situ. So if we take our bottle incubation, we've isolated these microbial processes. If the growth in our bottle matches well with the changes in situ that we're seeing, we know that these microbial processes are really dominating what's happening in situ. If on the other hand, the growth in our bottle doesn't match well with the in situ changes in phytoplankton biomass, we know that we have other loss processes and other processes in general that are very important in the system, say like advection or sinking, that aren't captured in our 1 liter bottles that we're incubating on board. And so the cruise design made this a unique opportunity to compare our results with in situ, but it's also the unique opportunity because there's so many amazing people doing so many measurements on this whole system, from the physics and the chemistry and the biology. And so I just wanted to put this picture here that gives you a little bit of a snippet of how many people have been involved in this work. And that's really made a unique opportunity to connect our rates to the biogeochemistry and to export as a whole. And as Dave said earlier, exports had 2 field campaigns, a low end member export region. So in our ocean station Papa, where we have this retentive food web, low export. And then we had our high export end member in the North Atlantic following a spring bloom. So I first want to share with you some of the results from the North Atlantic. And a quick note about our methods. We use a dilution method to measure phytoplankton growth rate, and then the microzooplankton grazing rate on that phytoplankton growth. We incubate our bottles on board in deck board incubators, and we have vertical resolution to 1% par, but the data I'm going to show you is integrated throughout the euphotic zone. So in the North Pacific, we see that our bottle experiments really match well with what's happening in situ. So we're capturing a lot of the in situ dynamics in these microbial dominated bottles. So here on this graph, we have our in situ integrated chlorophyll to the 1% par. And then these blue arrows, blue triangles, show what our predicted concentration of chlorophyll would be given our phytoplankton growth rates and our microzooplankton grazing rates. So you see overall that they match well with in situ, and we see that phytoplankton growth is balanced really well by microzooplankton grazing. And overall, on these 30 days that we're out at sea, we see a fluctuation of chlorophyll, but it's kind of around the steady mean. So in the North Pacific, we see the fate of phytoplankton is all being assimilated into that larger microbial and food web dynamics. And so, as I said earlier, this is really just a tiny piece of the whole puzzle. And I know this is an overwhelming graphic to just throw up here, but don't worry about the arrows and numbers to look at right now. I just want to show you that there's been a work done by 30 more people and the data from many more people in exports to really constrain this upper food web processes. And so this is in a paper that's in review in that Elementa Special Edition. And we have our depiction of the carbon flowing through the food web. And so just to simplify here, what our major processes are, we have our net primary producers making our organic carbon in the system, and then it's being consumed and it's well balanced by our microzooplankton grazing. And then on the other side, we have our bacterioplankton taking up the DOC. But in this retentive and high recycling system, we have much of our organic carbon then lost to respiration. And that means that the carbon that's available for export is byproducts of these large food web processes. So we have a bit of POC production from our mesozooplankton, and then POC production in the form of fecal pellets from our microzooplankton and detrital signals that contribute to this low export signal that we're seeing in the North Pacific. Just a graphic here that shows that because we know that microzooplankton grazing is eating so much of this daily phytoplankton growth, we know that the active export pathways in the North Pacific are those that are mediated by our zooplankton friends, and that these are largely inactive pathways of physical mixing, the sinking of cells and sinking of aggregates. So we see our carbon being shuttled into the food web and then portions exported in the North Pacific. And that's in really high contrast to what's happening in the North Atlantic. So in the North Atlantic, we hit the peak of the phytoplankton bloom and then the decline. And we see that our microzooplankton grazing is only eating a portion of the phytoplankton growth that happens every day. So in this graph, we have our time on the x-axis and the percent of growth that's grazed by our microzooplankton. So at the beginning of the cruise, we only see about 25% of daily growth being consumed, and then up to 75% as we go through the cruise. And so again, taking our incubation bottles and comparing that to in situ, we see that we would expect that integrated chlorophyll should increase over time because we're not getting all of the carbon consumed by our microzooplankton. So it would increase more rapidly at first and then more steadily after that 17th of May. But what we actually see is a large decrease of chlorophyll in situ, so a 60% decline. And so that means we need alternative loss processes for this phytoplankton growth. One of those processes could be mesozooplankton grazing, but we've done some experiments to show that they're really playing more of a top-down role on the microzooplankton. And so in the North Atlantic, we see that there's a smaller assimilation of phytoplankton growth into the food web, which means our active export pathways are these that export phytoplankton directly. So mixing, sinking of cells and sinking of aggregates. And so a quick summary, in the North Pacific, we see highly retentive and then export that is food web byproducts. In the North Atlantic, we see this export of phytoplankton material more. And so just to recap, that microzooplankton grazing is offering us this opportunity to get to that predictive understanding of where carbon is going and how to link these upper ocean processes to export. And with that, I want to thank the team and then also point you to where to find some publications that are coming out about all these pieces of the export campaign. Thank you.
Questions for Heather as Jessica comes up and gets ready?
Thanks, Heather. This is Charlie Stock from the Geophysical Fluid Dynamics Lab. I was curious about the last point you made about the mesozooplankton acting more as a top-down control on microzooplankton, and I just wonder if you could comment on what the implications of that for paradigms of mesozooplankton, the timing their diapause to consume spring blooms are.
Let's see. So yes, we did some experiments with some copepods with Deb Steinberg and Amy Moss's group to look at this effect of the mesozooplankton and top down. But I think the link to diapause and to the export pathways that way, I think Deb might be a better person to answer that question.
Yeah, good question, Charlie. You know, we didn't do, except for what Heather just said, we didn't really do grazing experiments on these cruises. So I don't really have an answer in terms of what the diapause copepods, at least in the North Atlantic, are doing. We actually didn't see a lot of Calanus and Marchicus on our cruise. I can say at the time of year in the North Pacific we were there, the neocalanus, which are the diapause copepods there, were not eating or pooping. They were just about to get ready to go down to depth. So they weren't doing much of anything, which is also sort of an interesting story because they were the huge biomass of copepods there, but they weren't doing much because they were ready to go to sleep, essentially. Yeah, but it's a great question, and yeah, something we can. Oh, good. Well, we're happy to help.
All right, and next we're ready for Jessica Liu, who is joining us from NOAA's GFDL.
All right. Thank you so much to the organizers for this invitation to speak. You heard some great talks already about gelatinous zooplankton, salps, so here are just some pictures of these beautiful pelagic tunicates on the right here. And then you also heard a great talk from Charlotte this morning on our system models and constraints on the biological pump. So here I will attempt to put some of these together and tell you about a couple studies on modeling pelagic tunicates. So ecological and biogeochemical constraints on pelagic tunicate carbon fluxes. So because I only have 10 minutes, I'm going to start with my take-home points. There's a common conception amongst biologists, myself included, when I started this, that our system models are missing some X organism or process. We anticipate then from empirical data or models that X contributes some Y petagrams of carbon per year to the biological pump. Therefore, global models are underestimating the biological pump by that amount Y. However, what I find, so I started with gelatinous zooplankton, particularly these tunicates. We find that from empirical data or models that tunicates can contribute 1.3 to 3.9 petagrams of carbon per year to the biological pump. And I'm going to talk about this study from 2020 in global biogeochemical cycles briefly first. But after we incorporate these tunicates in a global model, carbon export only increased by about 0.1 to 0.2 petagrams of carbon per year. But before you all get mad at me, I just want to say that we find that tunicates do produce a substantial amount of detritus, about 1 petagrams of carbon per year, but this causes a redistribution of export production away from the other zooplankton sources. All right, so if you missed the rest of my talk, that was it. So in this first study that was published a couple years ago, we did this data-driven model to estimate gelatinous carbon fluxes. We started with the biomass of, here's the biomass of pelagic tunicates, primarily salps. And we assume that this kind of represents this kind of a steady state, though we know that that's very much not the case, but kind of a steady state annual biomass. And then we create this model that estimates from a set of empirical studies relating body carbon to respiration and clearance rates and excretion, then the amount of carbon that flows to these various sources via reproductive loss and exudation and respiration, and then going to predators, and then finally some amount that goes to POC. And then from the data, we use the observed sinking rates to then estimate the amount of carbon that fluxes to these various depths, to 100 meters, to 1,000 meters, and then to the seafloor. And the results from this is a lot of numbers, but actually just like the main numbers are here. From this model, we estimate that pelagic tunicates could export, between 1.3 and 3.9 petagrams of carbon per year. There was, quite a bit of uncertainty quantification that we included here. And because they sink so quickly, this then results in about 5-fold increase in transfer efficiency to depth compared to just like kind of a standard, 100 meter per day sinking speed particle. However, these numbers are quite big, and from what you remember in Charlotte's talk, the total export is only estimated to be between 5 to 12 petagrams of carbon per year, so this would have been quite a substantial fraction. And this is because there are no ecological or biogeochemical feedbacks in this data-driven model. So we did that next. This paper was just recently published a couple weeks ago in Progress in Oceanography. And what we did is, again, with the cobalt model, I didn't fully explain, but the previous model kind of used some outputs from cobalt. But here in the cobalt model, there's 3 sizes of zooplankton, small, medium, large. There's 2 sizes of phytoplankton and a diazotroph. And then we added, so here's the kind of representation of the ecosystem, and then we added basically an appendicularian and then a salp or pyrosome, basically a salp doliolid pyrosome. And I don't have enough time to get into the details of the model formulation, but basically they're able to feed on small-sized particles, and both the appendicularians and the salps produce export, and they're fully embedded in this food web. Again, we use some data constraints, so we did another data compilation because the first data compilation didn't include appendicularians, and we used a number of different databases, as you can see on the left, to pull together a global map. And one of the things that we observed in this data is a really kind of interesting relationship between surface chlorophyll and biomass of tunicates and mesozooplankton. So we found that there was, well, a lot of spread in the data, but they're contrasting to the mesozooplankton. There's a relatively flat scaling slope for the large tunicates, suggesting that as chlorophyll may decrease, then the relative biomass of pelagic tunicates would not change that much. Then after putting together this model, we saw that the model roughly captures the mean observed biomass. The model points are in the black, and as well as this large-scale emergent relationship. There isn't an exact one-to-one match, but considering the spread in the data, we felt like this was a reasonable representation of these pelagic tunicates. And I'm talking about constraints here. So here I'm just going to show, this is what we have to do when we create new things, add new things into biogeochemical models. We have to validate them against observable quantities such as chlorophyll, nitrate, phosphate, and silicate. So you'll see that these actually are not that different between the now I'm calling GZ cobalt and the cobalt control, and that's actually exactly the point, is that we need to have any new addition to the model not degrade the surface nutrients and the surface chlorophyll compared to the observation. And so then what we find in the export is, so here I'm showing essentially a pie chart of the different sources of the export that goes into export production in the top 100 meters. We find that the tunicates contribute about 1 petagram of carbon to the export, but when we compare the total export production, actually doesn't change all that much. So in the control run, in the cobalt control, I have about 8.7 petagrams of carbon, and in the new model, it's just 0.2 petagrams more. But what it does do is it causes a redistribution of the sources of export. So, for example, you can see that the microzooplankton contribution, this peach color, has decreased. Same as this small mesozooplankton contribution. So really, when we add something new into the model, we can't think of it as just this addition to the modeled export, but rather when we put it in, there's all sorts of ecosystem interactions that result in a shift in the food web. All right, so back to my take-home points. Really quickly, we also looked at this, this detritus production. This is actually, my point here is not that they're totally insignificant. This is the summer detritus fraction. I kind of compared this with some results from Karen Samieskin and Debbie Steinberg. We get about 20% detritus production in kind of this North Pacific Station P area. And then when you look at the shift in the EZ ratio, which is export flux over MPP, you see that there is an increase in the relative amount of MPP that goes to export flux. And And I will end with this, that in Stephanie Henson's paper in 2022, she suggested a couple of feedback loops, positive, negative, and uncertain. And one of the ones that she suggested was that as a positive feedback loop, that increased stratification would result in decreased phytoplankton size and decreased POC flux. And I would say that actually this south feedback is a bit of opposite from this positive feedback that she suggested, where decrease in surface nutrients may actually result in an increase of POC flux as a fraction of MPP, and this could be actually a negative feedback loop. So that's it. Sorry for going over.
Okay, question for Jessica as Grace gets teed up.
Hi. Oh, wow, this is loud. This is a great talk. So question for you. I mean, earlier we heard that it's computationally expensive to add any of these components, but they obviously do, I understand completely that's not going to magically increase the export for 10 tons, but it does offer you a different view of the ocean in the same way, as we're getting closer to these digital twins that everybody's talking about. I have to put a sexy word inside, but how computationally expensive was to run cobalt, your experiment with and without tunicates in there?
It wasn't too much more computationally expensive. So in this run, I just had 2 new tracers for the new tunicate groups, but I actually have a run going right now where we added a fast-thinking detritus. So with all of that together, it's 5 new tracers. So it increases from 33 to 38 tracers. Essentially, I would say it's about a 10 to 15% increase in cost. Yeah, so we probably wouldn't be able to run this for 1,000 years, but we can certainly do order 100-year studies with this.
All right, we're ready for our next speaker, Grace Saba, who's joining us from Rutgers.
Hi, can you guys hear me? I'm here to add another X and Y for your models. That's what I'm tasked to do, and I don't have any Tic Tacs, so don't get excited. The video you just saw is the School of Menhaden off the coast of Long Island in New York, and you can see it's very immense. It's a drone footage showing this giant school of fish, and now imagine they're all pooping, right? So there would be a lot more fecal pellets, though, in the school of fish. So that's what I'm tasked to do today, is kind of convince you that fish are important contributors to the biological pump. And I am an ex-graduate student of Debbie Steinberg, so it explains a lot for what I'm doing here. Okay, so the biological pump, the early depictions are very plankton-centric. There was a lot of people admitting that today. So this is one of the first, I think it is the first depiction of the biological pump from the US JGOPS program in the late 90s, 2000s. And this is not the latest version. It's okay. I had an image of the biological pump that Dave showed before. Yeah, that would be it. Yeah, sorry. Okay, I'm restarting. Okay, here we go. All right, here we go. This is a depiction from today. I'm not naming any names or anything, but there's no fish in there. So fish have been generally ignored in our present day carbon budgets. We don't see them a lot, and we definitely don't see them in a lot of our modeled export budgets. But this is due to the very limited number of studies that we have on fish. Less than 10 studies have looked at active transport of mesopelagic fish, and they range from 1 to 20 gigatons of biomass in the ocean. It's a very large range. That's another big uncertainty with the fish. But we know they are very abundant in biomass in general in the ocean, and they migrate from the surface, between the surface and down to depths in the mesopelagic zone to about 400 to 1,000 meters. So very significant with active transport. And then only 5 studies have actually focused on direct measurements of the passive particulate flux, so they're fecal pellets. So very, just a handful of studies we have on fish, and that explains why they're not really incorporated. So OCB funded a fish carbon working group. I think it started in 2018. It's been a while now, but we pulled together a group of experts from all around the world to kind of just start talking about fish and their potential role in carbon flux. We were tasked with, or we tasked ourselves, I guess, with synthesizing the existing research on fish carbon flux, so what was out there, recognizing what the challenges were, like why didn't we have a lot of measurements, a lot of the methodological issues we have. And then we kind of went forward to develop some research priorities to start filling some of those gaps. And our paper came out, this is a product of the Fish Carbon Working Group, came out in Limnology and Oceanography last year. It's a very dense review paper, but we were very happy to get it out into the world. So what we did first was we summarized potential fish carbon sources. Where's the fishing pole? Here we go. Or bamboo. I guess it's bamboo, not fishing. Okay, so very similar role to zooplankton. They're producing fecal carbon, particulate organic carbon. One difference from zooplankton is that they're actually form carbonate. This is through an osmoregulatory process they have that because they're actually drinking seawater, they have to remove the calcium and magnesium that they're drinking. So they combine that with bicarbonate and excrete it as calcium carbonate minerals. And so that it can be incorporated in fecal pellets, and those are sinking as well. And they're excreting dissolved organic carbon, but they're also vertically migrating and doing all of those things at depth. So, and they're also dying. So fish falls are a big carbon producer that sinks down the ocean, down to the bottom depths. So a lot of different sources of fish carbon. So what we started doing was reviewing the literature, what we found published already, reporting estimates of passive and active fish carbon flux. And we found a lot of significant contributions from fishes. And this is on the left here, we have just one example of some coastal epipelagic. So northern anchovy producing these big fecal pellets that sink really rapidly to hundreds to over 1,000 meters per day. And then there's been some work looking at the active transport of fishes. So their fish are feeding at the surface, they're going down during the daytime, and they're respiring and excreting down at depth. And that's, this is just from one study that found about 32% of respiration from fish contributing to total carbon flux. And then carbonates are another significant source of carbon from fish. Still a lot of questions, but we do know that fish contribute up to about 45% of total surface ocean carbonate production. This is early work, but there's been some more recent work kind of looking to see if this is consistent across marine biomes and different types of fish. And it seems to be that it is a pretty consistent feature within fish. There's still a lot of unknowns with regards to what the solubility is of these particles, how much total alkalinity are they releasing basically into the water as they're descending, and how much is actually making it down to deeper depths. So what we did was we took all of those studies together, and we kind of just did a back of the envelope calculation to figure out how much of that carbon, how much of the fish carbon contributed to total carbon flux. So we had to have studies that were measuring fish carbon, but also measured total carbon flux through traditional methods like sediment traps. And the estimate we got was 16.1% of total carbon flux was from fish. And this is out of the euphotic zone with a very large error, as you can imagine, trying to take a handful of studies from all different parts of the world with different seasonalities and different organisms. And then we applied that to the modeled estimates of annual global carbon flux out of the euphotic zone, and that was about 1.5 petagrams of carbon per year. So again, pretty equivalent to what zooplankton do. It sounds almost equivalent to what Jessica mentioned with the tunicates. So that's another x to the y. Okay. And then we ask, well, okay, and Dave talked about this morning, is, okay, yeah, it's sinking out of the surface layer, but then is it being actually sequestered at deeper depths? And so the export depth meaning just out of the surface zone, and the sequestration depth meaning kind of going past the permanent thermocline to where it's sequestered for longer periods of time. And I do want to just point out that this is one of the first biological pump depictions that include fish, so yay. Okay, so John Dunn at NOAA GFDL did a ventilation timescale analysis using model-generated values of the annual global carbon flux at different depths, 100, 400, and 1,000 meters, and found that at about, from what is being exported out of 100 meters, about 34% of that is making it to 400 meters, and then about 13% of that is being, has made it past 1,000 meters. And the sequestration times corresponding to that are about 100 years past the 400 meter mark, and about 350 years past the 1,000 meter mark. And this is going to be more significant from the mesopelagic fish that are actually migrating to depths at, up to 1,000 meters, sometimes even beyond 1,000 meters. So they're really going to be the group of fish that's going to be contributing mostly to that really deep export. But that being said, the fecal pellets, even from fish living at the surface, sink very rapidly. Okay, so I was tasked to talk about the knowns of fish, but it is a known fact that we have a lot of uncertainty. So I'm just going to quickly mention kind of the things that we went through, and that was really estimating biomass of fish is really hard to do. They're just notorious for being difficult to sample, so we don't have good biomass estimates, and that's really significant for our mesopelagic fish community. Sampling passive flux, the methods we have tend to miss fish. They're very patchy, even more so than zooplankton, I would argue. They move a lot faster, so we're not really capturing that flux in our traditional methods. We don't have rates of like fecal pellet production rates, things like that, to even incorporate into models to figure out how much they're actually producing. It's really hard to keep fish in the lab, even fish that live right next to us in coastal waters. My graduate student can attest to that, and if you see their poster, you will hear from them. So we need indirect techniques needed to estimate rates, and this is particularly true for the mesopelagic fish that are nearly impossible to keep in the lab. So I will just hang on this one a little bit, and this is, again, work that my graduate student, Lauren Cook, has been doing most in the last 3 weeks. They just finished a big field effort there at Rutgers, looking at menhaden, and we were really trying to see the different carbon sources coming from an individual fish, which we haven't really seen yet, to try to get a carbon budget. And we were looking at things like respiration, excretion of DOC, egestion of fecal pellets, and there's a picture of the fecal pellets that we collected, and then some carbonates. So we can see some carbonates on the filter, and just some very preliminary results. The sinking rates were very fast. We're thinking diet is an issue, and they may not really be that fast. We are supplementing them with feed, which probably made them bricks going down. So we've got some troubleshooting to do. But we did see pretty high fecal pellet production rates, as this is one of the first measurements of fecal pellet production rates, so we were just happy to get some initial measurements there. And they conducted most of that. Oh, is that my timer? They conducted most of that at most of the, fecal pellets they produced were in the first 30 to 40 minutes. So we need fish-based carbon contribution to be understood so that we can improve parameterization of that component to the biological pump. We've talked a lot about models. Obviously, they need to be incorporated into models. I will argue that they do. I don't know how it's going to be done. We'll have to remove something from something else, but they need to be incorporated because fish seem to be pretty significant contributors to the carbon pump. And we also need to evaluate their role The role of harvesting, I have an asterisk by that, and I'll talk about that in just on my last slide. The role of environmental factors and climate change on fish carbon flux, and there was some discussion about that with Jessica mentioning Stephanie Hinson's latest paper as well. So how to get involved, we have a workshop coming up, and it's an OCB-funded workshop that's going to be focused on fish, fisheries, and carbon. So really just trying to figure out, so there's, ever since we kind of realized that fish might be an important contributor, there's been a lot of discussions on, well, do we need to protect all the fish now so that, you know, they're not going to take carbon away from export? And so we don't want to go there, right? We want to support sustainable fishing, but we also do want to just talk about, are there ways that we can make fishing more sustainable in order to preserve their role in the biological pump? So this workshop's it's gonna be virtual. It's a 3-day. It'll be split between a certain amount of time. We're still planning, obviously, but it'll be in the early next year, either spring, probably spring of next year. And then another way is to join JETSON, which is the Joint Exploration of the Twilight Zone Ocean Network. They do monthly webinars, and there's some really fun talks. There's some mixotrophy talks. I think it's a mixotrophy crowd, but they have a lot of talks, and they have a nice data base where you can kind of find data related to the mesopelagic zone. And then the UN Ocean Decade now has a community of practice focused on fisheries and blue food. And I'm looking forward to seeing kind of what activities they're going to put out soon, focus on that. So that's it. Thank you. And I do want to thank my Fish Carbon Working Group that worked with me on all of that information about fish carbon. So thank you.
Questions for Grace as Flavia gets teed up. Andrea, your hand shot up.
Hi, Grace. Andrea, Fassbender, AOML. That was really interesting. I had a question about the packaging of PIC and fecal pellets, and if you had the chance to think about how that might contribute to your reseed dissolution above the lysocline for calcium carbonate, stuff like that.
Thanks for the question. I don't know the answer to that. A lot of that, like Rod Wilson's been doing a lot of work on that. But the carbonates, so what we don't know about the carbonates is that we're starting to learn more about their minerality, and that's going to help us kind of figure out their solubility. So the questions really are, you know, they're coming down the water column. Are they being dissolved really quickly? I'm sure there's different ocean conditions that's going to cause them to be possibly dissolved faster or slower as well. And then, you know, if they're sinking down, are they getting to at least past the point where they're at least removing some carbon from the surface, or are they just being dissolved in surface waters? So I can't, like, we don't have a lot of information yet for me to be able to, like, completely answer your question, but those are kind of the main things that we're thinking about in terms of the importance of carbonates. Is basically that it really comes down to the solubility of them and the amount that they're producing. We don't even know an absolute amount of carbonates that fish are producing yet.
All right, now we're ready to hear from Flavia Boscolo-Galazzo, who's joining us from Bergen.
Yeah, thank you very much for having me here. I'm actually very happy to kind of be here and bring you like a paleo perspective on the biological pump. So basically what I'm gonna do today is telling you about like the biological pump in a warmer ocean, and I will show you a little bit how we combined policy record and from modeling results to reconstruct how the biological pump functioned in the past, and then how we used a similar approach to try to predict how the biological pump may function in the future. So this work I'm presenting today was part of my previous postdoc at Cardiff University and has involved a team of people besides the PI, of course myself, who developed the proxy component of the study, and a team of Earth system modelers, Catherine Crichton, Andy Ridgewell, and Jamie Wilson, who developed the modeling side of the project. So I'm going to start with something that may sound like a spoiler, because I'm going to say that this talk won't focus much on the export production term of the biological pump, but I will rather focus on the flux of organic carbon settling through the twilight zone of the ocean. Here, a lot of this carbon gets remineralized, gets consumed, and therefore what actually makes it, how much carbon makes it to the bottom of the twilight zone, is key for ocean carbon sequestration. So in particular, what I'm going to try to address with this talk is whether this organic carbon flux through the twilight zone could be even further reduced in a warmer ocean due to temperature-dependent remineralization processes. So we came out with this paper in Science last year, where we tried to address this research question by looking at warmer oceans of the past. So we started looking at the strength of the biological pump from the middle Miocene, a time interval when the ocean was about, the middle Miocene 15 million years ago, when the ocean was between 6 to 80 degrees warmer than today, and then we moved on to the following cooling trend, which led to modern days ocean temperatures. Focused on a range of globally distributed IODP sites, and we used a group of Vicarious zooplankton called planktonic foraminifera. We used their fossil record as tracer for organic carbon remineralization rate. In particular, we measured delta C13 on their fossil calcareous shells and used delta C13 measured on surface dwelling species and dwelling species to reconstruct the delta C13 gradient for the upper part of the water column, and use this delta C13 gradient to track changes in organic carbon remineralization rates through time. Then compare this delta C13 gradient with the same parameter from an Earth system model with and without temperature-dependent remineralization processes. So this plot summarizes some of the main findings of the study. What you can see here is the reconstructed organic carbon remineralization rate, these black stars, plotted versus the modeled organic carbon remineralization rate. Here, remineralization rates are expressed as rate of delta C13 change. That's for the upper part, for the upper 200 meters of the water. So two main findings emerge from this plot. First of all, over time and with cooling, you need to go slightly deeper into the water column to find the same amount of organic carbon remineralization. And second, reconstructed remineralization rates are basically overlapping with those modeled with temperature-dependent remineralization processes, and very different from those modeled without temperature-dependent remineralization processes. So this comparison between data and modeled organic carbon remineralization rates argue toward a role of temperature in controlling organic carbon remineralization in the water column over geological timescales. So then what's the implication for the biological pump and POC fluxes? So here I'm showing the modeled POC fluxes obtained with the temperature-dependent model, which pretty much shows a 20% decrease in export production over time and with cooling, which is nonetheless counteracted by a more than doubled transfer efficiency at twilight zone depth. So what we are seeing is that basically moving from a warmer to a colder ocean, the biological pump becomes more efficient in delivering organic carbon into the deep ocean because of lower rates of metabolic activity in the water column. So we also find that this increase in the efficiency of the biological pump over time and with cooling had an impact on the evolution of twilight zone plankton. So we reconstructed the paleodact habitat of planktonic foraminifera in the water column, and so that over time and with cooling, more and more species evolved in the twilight zone of the ocean, with colored symbols. Also, the abundance of these deep-dwelling species increases over time. So what we think is that increased food availability into the deep ocean over time and with cooling due to this increase in the efficiency of the biological pump acted as a booster for evolutionary rates in the deep ocean. And we don't find this only in planktonic foraminifera, but also in calcareous nanofossils, which is a group of mixotrophic algae, which also show this increase in deep dwelling species over time. So this could be an evolutionary trend shared by many twilight zone organisms, which differently from calcareous plankton don't have a fossil record to show it. So next, we look at warmer ocean of the future. And basically, we use our temperature-dependent model to try to predict what could be the behavior of the biological pump in the modern ocean and in the future ocean. So for the modern ocean, we use historical forcing, and for the future ocean, we use 3 CO2 forcing scenarios, in low one, an intermediate one, and a high emission scenario, which we applied up to here 2300, and then let the modeling run for several thousand years after. And you can see here as an example, the response of sea surface temperature to the forcing for the low latitude and the global ocean. So how the biological pump responds? So here is the POC delivery at 600 meters depth, and what we see is that we already seen a declining of this flux by model here 2020, and then this flux is projected to basically halve by the mid of the next century for both the intermediate and the high emission scenario. So what could be then the implication for the twilight zone community, which we just seen appears to have evolved over the past few million years in response to a more efficient biological pump? So we kind of did an experiment, and we tried to use the paleo distribution of planktonic foraminifera from the fossil records to try to predict the behavior or the future twilight zone plankton community. What we did is basically using the paleo depth habitat distribution from the middle mines into the modern of planktonic foraminifera and the abundance of deep dwelling species over time. We linked the abundance of planktonic foraminifera in the twilight zone with the POC flux in the twilight zone through a linear regression, and use this linear regression to project future changes in the twilight zone community. And these are the results. So here you see the projected change in the twilight zone community for each of our 3 CO2 emission scenarios. And what we see is that a decline in the twilight zone community could be already underway, like about a 10% decline by model year 2020. And then what we see is a 40% decrease by the mid of the next century already for both the intermediate and the high emissions. The abundance continues to decline for the following centuries, and we don't see a return to pre-industrial conditions for the whole duration of the simulation. So to conclude, the biological pump behaves differently under different climate states. The transfer efficiency is weaker when temperatures are overall warmer, and is stronger when ocean temperatures are overall colder. We found this linkage between temperatures and food availability at depth, which acted as an evolutionary booster for the twilight zone plankton community over the cooling trends of the past 15 million years. Vice versa, if current ocean warming continues into the future, it's likely to weaken the strength of the biological pump and to represent a threat for the twilight zone plankton community. And this will involve a wide range of organisms, such for instance micro and macroplankton, twilight zone fishes, and many other deep sea creatures we know little about. And thank you very much for your attention.
Questions for Flavia as we get Tristan teed up?
Xinping Hu from Texas A&M University Corpus Christi. So in the correlation, you show this 4M and POC flux correlation is this assumption, right?
It's an experiment, I would say. We try to use the distribution, like the abundance from the fossil records, and then we link it with the model POC flux at the same depth, and see basically what it gave us. Obviously, yes, assume that the modern twilight zone would behave similarly to the one in the past, yes.
I see, thank you.
All right, right, and we're ready for our last talk of this session. Welcome Tristan Cordier, who's joining us remotely from the University of Geneva.
Hi everyone. So yeah, I'm very grateful to be given the opportunity to present this work remotely. So yeah, actually I'm now a senior researcher at North in Bergen, Norway. But the work I will present today is great. It was mostly done at the University of Geneva, where I did a postdoc before Bergen. So we spent some time in the surface. Now we just came to the twilight and the dark zone, and I will dive even deeper to reach the seafloor in this presentation. So in which I will present the work that I'm doing on sedimentary DNA and how I plan to use that to try to reconstruct also the past functioning of the biological pump. So some words about DNA. So every living organism has DNA, and it is used to reconstruct the tree of life. So based on the specimen sequences, when you extract the DNA from an organism and you compare it to existing database, you can say more or less which species or which taxonomic group at least it belongs to. But when you do that from an environmental sample, that can be sediment, that can be water, hair, some feces, or pellet, you can also, that falls under the umbrella terms of environmental genomics, in which you extract the total DNA of the samples and you try to make a community profiling based on this based on small sequences. So a brief overview of the workflow. So I said we take an environmental sample, so we get the DNA from both living organisms, but also for the ones that are recently dead, or even extracellular DNA, or DNA that is attached to particle. So we extract the total DNA content of these samples. We amplify by PCR the whole eukaryotic communities. We don't target only, we don't target COVID, but that's the same idea behind. And then we massively sequence all of these molecules that we amplified with the PCR. Then there is some bioinformatics involved to curate the data, to cluster the reads into taxonomic unit on which you can assign a taxonomic label. And then that's what you use to analyze your data and to test your hypothesis. So of course it has massively boosted by the boom in sequencing technology. I won't get into much detail, but the overall idea is that we produce more and more data and much longer fragments of better quality. And just in a decade, for example, here is the first metagenome that has been published in 2004 for water content, and 10 years after there is the Tara Ocean expedition that produced more than 500 times more data at a cost divided by the 1,000 ratio. So that's then you can envision to deploy these methods at global scale, and that's basically what the Tara Ocean did in their expedition. That on these fetal sailing boats, they sailed all over the world over 4 years. That involved one ship, and they mostly focus on the surface ocean. With plankton nets and CTD sampling. And they produce genomic data for the full size fractions of plankton, from viral particle up to large size animals. Then another expedition that occurred at similar time is the Malaspina expedition, a Spanish expedition. And over 2 years with 2 ships that involved more gears and sampling and infrastructure, and they focus mostly on the twilight zone and the meso and bathypelagic zone of the ocean, and they focus mostly on the smaller size fractions, so that microplankton, anything below 100 micron, basically. And here, this is a team of deep sea biologists with whom I had the privilege to work during my postdoc in Geneva. And so over the years, they collected sediment, deep sea sediment samples at global scale too. And so that involved more than 10 ships and over 16 deep sea expeditions. So that's probably the most expensive mud you can get. And that's in deep sea floors, it's teamed also with microbial life and strange animals that we basically don't know much about. But when you get some deep sea sediments like that, one of the challenges when you work with DNA is how you can separate a signal from sinking plankton that finish via aggregate on the seafloor from the indigenous benthic biodiversity that is living, feeding there, reproducing there. When you get multi-core, when you get mud coming on the ship, you have no way to do that, of course. And even if you would sequence, you have no way to do that. But since I presented these two expeditions in the water column, what we did is to actually sequence the very same genetic marker for our sediments than the one used for the water column. And by directly comparing the 3 global scale data sets, we were able to separate the DNA signal that originates from the sinking plankton from the benthic eukaryote. And by that, I mean that it's not only, of course, if you find a diatom on the sediment, it's most likely it is the phytoplankton and it has a chlorophyte. So deep sea organisms wouldn't have much use of a chloroplast, but you have many groups that contain both planktonic and benthic organisms. So just based on taxonomic annotation, this is very not straightforward task to get into. So by that, with the DNA fragment, we're able to directly compare regardless of any taxonomic annotation. And one you can see here on this graph is that the plankton fraction in sediments includes most of the stuff we know, that we expect. So that includes dinoflagellates, diatoms, and also a lot of radiolarian and fungi and copepod, small amount of copepod. And what is striking is the vast component of completely unknown eukaryotic diversity that is specific to benthic organisms. But we will focus mostly on that now. So with that data, the 2 questions that I will dive a bit deeper into is what kind of pattern there is of plankton DNA reaching the deep ocean floor, and how we can use that data to link to the biological pump, and what can it tell on the functioning of the biological pump. And if I have a few minutes, I will give a snapshot on benthic eukaryotes. So when we check, when we analyze the abundance of planktonic DNA in sediments as a function of latitude, we can see that, as expected, there is a higher deposition of plankton at high latitudes. This is an expected pattern. What is maybe more interesting is that we have actually also the most abundant plankton taxa are the one that you can find on the seafloor, but there is also many very abundant plankton taxa that you don't find on the seafloor. That can be a sampling issue, but I think there's also a pattern here. And when you compare the functional group of the sinking versus the non-sinking plankton, you realize that there is a higher proportion of parasites. That's also, I think, an ecological expected outcomes, because once you have a phytoplankton bloom, this biomass can be utilized by other organisms, and then all of that sink to the seafloor. And as a function of latitude, we can see that parasite abundance is mostly prominent at a low latitude, so in oligotrophic water, which also, I think, is an unexpected result. So now when we use this data and we compare and we get the existing particulate organic carbon flux model. So this is here from the surface. So based on sediment trap and thorium isotope, this model calibrated on observation and reprojected at global scale. And that's from the surface at 100 meters. And in this work, using also remote sensing, they modeled the fraction of this export that is actually reaching the seafloor. So using this plankton DNA data set, I used random forest regression to try to see if we can, if the community profile that we get from the plankton DNA is a good predictor of the variation of carbon fluxes. And the thing is, we can explain about 60% of the variation of the fluxes just based on plankton DNA on the seafloor. That also means that we reveal some plankton taxa that contribute to long-term carbon sequestration in deep sea sediments. And not unexpectedly, we see a lot of diatoms. We see also some dinoflagellates, but what is interesting again with parasites is we have some marine straminopies that contains mostly parasites that show up as important taxa explaining the variation of the pump. So what I'm trying now to develop in Bergen is of course, given this result, it paves the way towards using plankton DNA to reconstruct the past functioning of the pump if you go on subsea floor sediments. And that's exactly what I'm trying to develop now. So based on this idea, using carbon model, the idea is to identify this biomarker, which I showed that exists, and to try to screen those in down-core sample. With this question in mind, will climate change strengthen or weaken the biological pump, and with any feedbacks that we are all aware of on the climate. So as a rapid test of this idea, I used the recently published V9 18S dataset, so that's the markers that we used for our study. And I just compared our surface sediment DNA plankton at global scale with this ancient plankton DNA. And here this is an ordination in which you can read that the closer to point R, the most similar the community are also. And what is important to note is that the ancient DNA is not that far. I mean, it overlaps to some extent to our reference samples. So this means that we can possibly project and reconstruct the carbon flux. And here, well, this is only a core spanning the Holocene, so the 10,000 years before present. And we see a decreasing trend in reconstructed carbon export over the Holocene in the west coast of the US. And that's consistent with recent work showing that glacial maxima are associated with the highest carbon sequestration in deep sea sediment. So with that, one question that I'm trying to tackle now is to try to reconstruct this carbon export on specific time scale. So that would focus on two glacial maxima, the last glacial maxima, and also focus on the last interglacial that occurred about 120 to 130,000 years ago. Because this is a climate, this is considered by geologists as a climate analog to ongoing changes. So we would compare the reconstructed carbon flux around that time, and that would inform us possibly on what we are about to see with climate change. So now, given this time scale, one question you all may have in your head is how far back in time we can go with ancient DNA. And I think this is a fairly open question. I don't have that answer to you, but what I can say is that we have Daniel Grant, a PhD student working in our group, that has this paper in preparation, in which she's studying a sediment core that span the last interglacial in the Eric Drift in the Labrador Sea. And she reconstructed the eukaryotic communities on this core, and we can see that we have data for the last interglacial and for also the MIS6s that correspond to the glacial maxima, taking, of course, many precautions to avoid any kind of contamination with modern DNA. And her result shows that the biodiversity shift occurred. I mean, this shift they align with climate transition. So that's also promising for the work I would like to do with the pump. But one limitation that I'm still thinking of is how we could possibly validate the DNA-based pump reconstruction. So one way I think to do would be to use a sedimentation rate, but I'm not a geologist, so any insight here from the audience is most welcome. And I think also with Earth model based reconstruction, that could be also a way to have, if it's consistent, that we would have then multiple line of evidence of at least trends in the response of the pump to climate forcing. So I don't know if I have time, but I will just do a very quick snapshot on benthic eukaryotes. So here I said that many of them are known, And just to give you a feeling of how we are blind to deep sea benthic diversity. So on this graph, you have on the left, the more on the left, the closer the sequences are matching something that we know, that we have a reference sequence in a database. And you can see that for benthic eukaryote, we are way behind the kind of standard for taxonomic annotation at the super eukaryotic group. So many of those sequences are totally new to science, and we cannot even say to which eukaryotic supergroup they belong to. And this diversity is specially structured in the ocean. So there is a lot to dive in there. And with that, I will thank you for your attention. I will take any question.
Thank you so much, Tristan. Questions? And I'm also going to welcome up all of our session speakers from the entire day to come sit up front for a panel discussion.
Hi, Tristan. This is Colleen Durkin from MBARI. I was really curious about how much does degradation impact your interpretation of the data in terms of like preferential degradation of different phytoplankton groups in the sediments or of different DNA? Like, do you have to take that into account or constrain that at all in your approach? It's something I've been thinking about a lot, so just curious what your thoughts are.
Yeah. Thanks for that question. No, we actually, I mean, for surface DNA, we don't. We assume that anything that reaches the sea floor is probably, anyways, the most. I showed that it is mostly the most abundant plankton that reach the sea floor. So I think that tells that there is many more rare taxa that are not even reaching the sea floor. They are probably consumed on their way down. But what I can say that in this data, we see also that overall the abundance profile in the water column is broadly preserved in the sediment. By that I mean when I take the full data set, so I take the full surface ocean abundance profile. What the most abundant plankton in the surface is also the most abundant plankton in the sediment. And that tells me that broadly the pattern are conserved. Then if your question is about the ancient DNA, I think this is $1 million question. I would love to know that. And I think it's a very new field, and I think we have a lot to discover and to get to know.
Thanks.
Take a minute anyways, go for it. So question for, question for Grace. So thinking about the fish is really interesting, and it led me to wonder, how granular do we need to get with all the other organisms that are then out there? Thinking in terms of squid that are accumulating in deep scattering layers and large euphausids, and it seems like we have the gelatinous organisms are being covered a little bit, and things smaller than that we're kind of getting a sense on, and then fish. What about those things in the middle? How significant are those likely to be?
Yeah, it's a good question. I don't think we have a lot of information from those organisms. We have some information from organisms that vertically migrate, because those will come up in the tows that they do, and sometimes those contain things like decapods and some small squids. But in terms of like actually studying their process rates and production of carbon, we don't have a lot of information on that, so I can't really answer your question, but I think the more we know about just their behaviors and their biomass and abundance, the better we can do to try to make some of those estimates. Maybe Debbie will have an answer to that, but I think she missed the question.
How granular do you have to get?
Yeah, I remember from a meeting we had at National Oceanography Center that squid are really abundant and really, I think, important, more so than we realize. So that's one group of organisms that I think we need to also pay attention to. Yeah, just add that. Yeah, I don't know what squid poop looks like either.
I'll throw out there, what do people think of, forgive me if I'm phrasing it wrong, but Jessica's suggestion there's one pie and it just gets divided up differently if we include more organisms. So do we have the potential maximum carbon export correct?
What is? Hey, I think there's only so much reduced oxygen in the ocean. That's to balance everything. There's only so much AOU. So if you build more and more piles, as Jessica showed, if you build more and more things, it just redistributes. I mean, that's just the way it's going to have to be. Because there's only so much that the biological pump can't explain, and we know what that balance is.
I also think that even if that were true, and that's actually how it is, it's important to be able to, and I think you agree with me, to know the mechanisms of which pathway it's actually going in. And that's important.
Yeah, yes. I think the source attribution is really important too, and what was intriguing about Tristan's talk is, is there evidence of squid or fishes in deep sea sediments that would indicate that these organisms are getting there? Because if we only see record of the plankton, then that organic carbon is making it down, but the fishes and the other organisms aren't. So I guess I had a question maybe to my co-panel member, which is, Tristan, do you see any debris of fishes or any organic remains that you could tie together with your DNA evidence? And do you see any discrepancies between, or any validatory evidence that kind of together forms a much stronger conclusion about the carbon that's making it to the sea floor?
Yeah, we have some data on vertebrate, but unfortunately I cannot go further down to the taxonomic annotation below vertebrate. So I know that we have quite some sequence of vertebrate, but I don't know if that can be, they are most likely fishes, but I wouldn't be able to say. And I cannot really relate, we didn't measure the organic carbon as such, so we just get the DNA. We just know which are the contributor. But from few grams of sediment that we use to extract the DNA, I would say that we mostly have the small microbe or the small plankton that can more spatially cover the seafloor. Fishes would more like make a bump or spatially structured, I imagine.
You want to go, Jessica?
Can I make a comment? Yeah. So to Susanne, yes, I did present it as a pie, and we're redistributing it. But that is very much only for the surface ocean. That's euphotic zone. Once we get down to the twilight zone, we really do not know what that. I don't even want to call it a pie. I don't think it's viewed like that, but we don't know what the transfer efficiencies are. We don't know what the remineralization rates are. So that's the big unknown.
If I could just add something about the burial of big things like, or finding eDNA of fishes or even squids or things like that on the bottom of the ocean, I would think it'd be very unlikely to see that, because if those things end up on the bottom, they get eaten, right? They're the organisms down there, scavengers that travel long distances to find these food falls, and they snap them up, they eat them all up. So I wouldn't expect to find a lot of that on the seafloor. Yeah.
Okay, let's start with this one. So this is from Seth Bushinsky in Hawaii. On the one hand, we have Charlotte's point that carbon export in models is ultimately constrained by nutrient availability, so adding or changing carbon export mechanisms doesn't change total export. Jessica's work demonstrated this as well. On the other hand, in order to predict future changes, we need to accurately capture mechanisms of export so that we can understand how they change. We've heard about a lot of different important mechanisms today. Does anyone have insight into how important the carbon export mechanisms or parameterizations of them can be included in Earth system models to improve our predictive capability for future changes? That was a long question.
Okay, I have something to say.
Can we parameterize any of the processes that we're talking about this afternoon? Is that the short version?
Okay, I would like to say something to that. I think if we think about what mechanisms we like to put in Earth system models, I think an important question is what for exactly? If you're really fixated on the question, how big will the global carbon export be in 100 years from now, then maybe many of the more detailed mechanisms that were shown today, it wouldn't matter too much in our present-day Earth system models. But I think that's not the only thing we want to know. If you are, like me, additionally interested in how the particle dynamics exactly work, where the particles come from, what that means for the ecosystem, what that maybe even means for fisheries, then I think, yes, we need this separation into those more detailed fluxes. Or also, if we think about the same future geoengineering ideas, then and we maybe need one day to be able to predict what this would do to ocean ecosystems. And yes, also I think we need those refinements. Just that it's really only the maximum global carbon export that's possible. That is really strongly tied to the nutrient influx. That we maybe don't need it.
Hi, thanks. This is Jamie Palter from URI. Really enjoyed all the talks. I guess this question is mostly for Grace, but maybe some others would chime in, which is, we've all heard about how humans have fished down the food chain and removed many of the big fish, like really at very high percentages. Has that provided a natural experiment of what happens when we, go to smaller organisms in the ecosystem? And is there any data on AOU or anything that could maybe be compared to the times of great declines that might give us a hint about how that influenced export, or any other data that might give a hint?
Hey. That's a really good question, and there's been a lot of discussion about that, and just trying to figure out if harvesting can impact, I guess, the potential for carbon to be sequestered by fish. And I think we don't know a lot about even impacts of size on carbon production rates. How much, if there's a size specific or a size spectra of that, I guess in terms of how much carbon is produced by fish. We don't have really any information on that. So it's hard to kind of go backward and say, less big fish means less carbon, because there might be more smaller fish with similar overall biomass that might even that out. And also, if there's loss of fish, there could be maybe more zooplankton, and then maybe that evens out in terms of total, carbon production from zooplankton and fish combined. So there's a lot of different, scenarios that we are talking about that don't really have data to try to get at. I don't know if anyone wants to add more to that.
Okay, I've got another question from Bob Anderson. In the late 80s and early 90s, there was a debate in the literature about whether or not the biological pump is responsible for any of the ocean's net uptake of anthropogenic CO2. Galen's response yesterday, and I think Charlotte agreed today, was not as far as we know. In contrast to statements made at this OCB meeting, my experience indicates that this issue is still not settled. This is an important issue that needs an authoritative resolution. What does the panel think? Bob's not even here, and he can still poke the bear.
Hi, Bob. Okay, we know that there's very little atoms that are anthropogenic of carbon that are part of the biological pump. There's just 38,000 petagrams of DIC and not that much anthropogenic carbon. So we know that. So, you know, Bob's not here to argue with, so sort of is. And this has gone back a long ways. We do know that if the biological pump is shut off, then we know that we've eliminated one of the main ways that reduces the surface DIC concentrations and pushes it. So that I think these are how many angels dance on the head of a pin sort of arguments. Sorry, don't mean to be a pinhead. This is this is a bit of a philosophical point. I personally know that they both have to be active. That's the way that it has to be. I don't know how productive this discussion is, and I probably said that 10 years ago, the last time that Bob and I had this discussion. But they both have to work in concert. I mean, I think we know that. But my opinion.
Not part of the panel, but I think the consensus was that the uncertainties are so large on the processes that it's hard to demonstrably say, this is the percentage, or here's the effect on the biological carbon pump. And so I want to encourage everyone to participate in the breakouts later to get us past this bottleneck.
Yeah, but Susanne, the issue has been that the biological pump itself does not move any anthropogenic. All agree about that. But the fact that if it's not there, then it makes the physical pump not act. I didn't say that well. Both have to be there.
Yeah, but now I'm changing my comment, actually. I was going to talk about how I'm afraid of SALPs, but instead, I wanted to talk about this comment because I think there is some evidence in the literature that would suggest in an ocean that is accumulating more carbon, you can have no change in the biological pump and you can influence air sea fluxes. And that is a very nuanced point in the literature that's somewhat recent, but I agree. I think it kind of depends on if you would argue that the added anthropogenic carbon, even if it doesn't influence, the biology, it does alter the fluxes. You have to argue, is that an anthropogenic thing or a natural thing? And I think that a distinction is needed there.
Oh, this is Julie Granger, UConn. Just adding to the confusion, I'm wondering if there were no biological pump, of course there'd be more carbon in the atmosphere, but would there have been less anthropogenic CO2 dissolved in the ocean?
There wouldn't be any humans. Yeah, there's. So Julie, I mean, if you didn't have a biological pump, how would you wouldn't.
You could still dissolve the anthropogenic CO2 in proportion, Henry's law, right? Which is the way it gets in there. It's like a general exam question.
Yeah. That was 40 years ago for me, so.
Sure, I don't want to change the topic if there's a response to that.
Oh, I was just going to say yes, but please, I'll ask a different question next.
All right, this is Holly Muller from UC Santa Barbara. One thing that I thought I picked up on maybe from the modeling side of the discussion was that perhaps a way to change the size of the pie, if you will, might be different stoichiometry of the material that's being exported. And I was wondering from either the modeling perspective, but especially the empirical perspective, under what circumstances we might expect to see higher carbon to nutrient ratios of the material being exported, which biological linkages, what seasons, et cetera, and how that might change in future climates.
I can offer one small comment to say that I think mixotrophs bypass some of that traditional stoichiometry, and so intriguing for me and the organism we studied that it doesn't respond to nitrogen. It responds more to phosphorus, and I would say even in vitamins is more its preference. So as we saw, dinoflagellates are getting to the sea floor. So I think they're an important group because they might have unusual mechanisms. I think, Debbie, you made the comment about the lipid pump for zooplankton. So I think there are certain mechanisms, and maybe they do point to areas where we've got to focus.
Also comment on that. For instance, temperature has been seen to alter, could potentially alter the stoichiometry of sinking detritus, because like phosphorus and nitrogen are preferentially consumed, and then therefore that leads more carbon, and then could potentially be like a negative feedback for the changes in the efficiency of the biological pump, for instance. So temperature also could be like affecting the stoichiometry.
Just one more comment. Many of the Earth system models have nitrogen fixation built into it, so they have ways to be looking at those sort of processes.
Brendan Carter from the University of Washington, and I've got a question for Jessica. If I understood correctly, the pie that you were showing was export, is that correct?
Yeah, export at 100 meters.
Okay, thanks. So also, if I understood correctly, you kind of added the tunicates, which were a more efficient exporter than maybe some of their competitors. So would it be fair to say that even though that pie didn't change, the overall production in the surface ocean maybe did change significantly? I apologize, you covered that.
The overall production of detritus just increased like 0.2 petagrams. So I would still consider that pie to be relatively not changed. It increased a little bit, and it increased the export ratio, the E ratio, for example. I think the interesting thing here is that there is a feedback loop that is different from the conventional accepted feedback loop that's associated with stratification. And for that reason, I would advocate that that would be why we would want to model these tunicates explicitly.
Gotcha, thank you.
Brendan, can I just add that if the remineralization scales, if they go deeper into the water column, that could have an impact on the sequestration due to those things, which is different
exactly.
For the same export.
Yep.
All right, Xinping Hu again from Texas A&M University Corpus Christi. So I come from the latitude with a lot of the hurricanes. Okay, so just to switch gear a little bit. So when we talk about the global warming and climate change, there's a discussion on this the nutrient, so that nutrient doesn't come up. Okay, so on one hand, that's one thing. So it reduces the production in the surface. On the other hand, when we talk about the global climate change and there's a stormy seas, so how do the panel, reconcile? Would there be a latitudinal shift in the production at all? Speculation, please.
No, I was trying to be quiet.
So the question was whether with warming we get increased stratification, but how might increased hurricanes affect that in the future? To balance each other out, yeah. I've just been reading about this when I was preparing for this talk, and I think some of the initial thoughts were that I don't know what the mass balance would be, but that hurricanes would certainly enhance export. But I don't know. Yeah, it's interesting to think about what would the frequency of hurricanes need to be to balance that out. And I can't answer that, but I think it's a great question. Probably some people here can answer that better.
No.
I was. Oh. No, it's.
Oh.
Okay, I was just going to add a fish perspective to that, that if the storms are occurring, if the frequency of the storms is occurring closer to like fall, like in the mid-Atlantic, for instance, that actually causes the fish to move offshore. They do the seasonal migrations, near shore, offshore. Most of the fish that are residing in the mid-Atlantic do that. And so if you're seeing an increase in frequency of storms, particularly, you know, hurricane season through the fall, these fish are just going to leave and go offshore earlier. And then so that possibly could transport carbon offshore from the shelf to the slope waters. Maybe, we don't know.
This is just a quick comment, maybe for Dave or other exports folks, but with reference to the stoichiometries we were talking about a minute ago, you know, the C to N to P and others are really important, but also remember the carbon to chlorophyll. And I think for the remote sensing stuff, that one is especially important, and the variability is still so high. And I know there's smart people working on this, but you know, it's something to keep in the back of our brain.
Can I make a comment on the hurricane? Yeah, if anybody else doesn't have a comment. I would actually suggest that it may not have that much of an impact for the purpose of and this is the first time I'm thinking about this question, so I'm just speaking off the cuff, that if a hurricane comes and mixes through a portion of the water column, then that portion will continue mixing, and it's kind of similar to this submesoscale eddy impact that Laura Splondy looked at a few years back, and I can't remember the exact citation of that paper. I think it was maybe 2020, where she found that there was overall not a strong impact from submesoscale mixing because it's compensated. There's like a compensatory effect from the upwelling and the downwelling.
Hi, I am Susanne, a postdoc at the University of Southern California, and I have a question, I guess it's for all of you actually. We spoke a lot today about the ecological sources, who is contributing to this POC flux, and there are many amazing contributions and the why of the question, but I was thinking about the other way around, right? What is contributing to the consumption of this POC and affecting the vertical POC flux? So like Charlotte said, that remineralization rates are poorly constrained. So if we think more about how bacterial consumption is happening in the ocean, and for example, there are recent studies that investigate the lability of the particle, the size of the particle, and how microbes will consume this particle. Perhaps they will consume faster in certain regions, but is lower in others. So is this something that we should focus on to kind of better constrain models? Are there measurements in the sea, like in kind of exports project measuring the rates, or I don't know, it's just a thought out there, bringing the bacteria component.
I'm happy to comment. So something that I had thought of in this morning's session was being, as biologists, I always like to sort of prod the system, and I was thinking about experimental water columns where one could explore factors involved in remineralization rates. I know there's a lot of processes going on in terms of sinking, but for some of those questions, are there useful ways that we could experiment and explore the nature of certain sources of particulate matter and how they would be remineralized? So yeah, I don't know whether anyone else has thought of these ideas, but is it worth exploring? I'd be keen to hear thoughts.
I mean, definitely bacteria are main drivers of remineralization. There's been like a recent study also showing that when like bigger particles get fragmented, that like facilitate the consumption like of small particles like by like free living bacteria that are not attached with the particle themselves. So anything that helps bacteria working and like facilitate their metabolic processes eventually is gonna contribute to remineralization. I think having a better estimate of flux activation through the twilight zone especially is gonna be important to kind of figure out which area of the oceans, for instance, have the greater rates of remineralization. Why is temperature, is something else, is the composition of the material. But for now, we just have so little information that even like constraining, for instance, like simple parameters, like even the physical role of temperature is going to be important.
I'll just add also that the work that we do resolving different particle types with depth, you know, what we found at the locations where we've done that and have multiple depths is that different particle types attenuate differently with depth. And so that's one way we can start to get at that. And then to your point of like thinking about who the grazers are, who's packaging that, you know, what we saw in that California current study that I showed you where we did the DNA sequencing, one of the kind of interesting results that came out of that I didn't highlight here was the small phytoplankton seemed to be preferentially especially found in the largest particles. And it seemed to be, I think those pellets were coming from some type of gelatinous zooplankton, maybe pteropods or maybe pyrosomes, but so it was probably from some kind of filter feeding, something that was preferentially able to eat small phytoplankton. And so the presence, I think that if there is a certain type of zooplankton present, that is going to have a huge impact on who ends up contributing to carbon export. So I think that the zooplankton are extremely important. Oh yeah, and the large cells tended to be in the small particles. So it was like this large phytoplankton were in the small fraction, small phytoplankton were in the large fraction. It was pretty interesting. And I think the zooplankton were mediating that.
Hi, Melissa from University of Hawaii. I was wondering if, with all the conversations about the CDRs and some of the approaches, I imagine that these communities, depending on the approach that they want to do, but for example, like salinity enhancement could change the communities of this species. So I was wondering, and maybe this is not the time to talk about it, but I was wondering your feedback about, like, do you feel comfortable? Do you think that we can model those changes, or I don't know. I was just wondering, what do you think as a biologist community?
So the CDR game involves a geoengineering influence to change how the ocean's working. It's either alkalinity enhancement, it's iron fertilization. There's a bunch of ideas. We need to know what the base state is. That's still, we've got to beat down the uncertainties we have. That's an important aspect of this. A few of us were at a DOE meeting, an RPE meeting on this last week. And one of the things that seems to have come out of this is the understanding that we really do need to beat down these uncertainties before we start doing uncertainties in how the biological pump functions before we start doing all this. So it is important, and not just for the alkalinity enhancement.
Sorry. Okay. Hillary Polewski, Boston College. So, yeah, I think the question that I've been thinking about sort of percolating through this section, there's often a focus from observationalists, and I certainly count myself as one, is thinking about all the things that we need based on our observations and all the complexity of the system that we want to put in the models so that the models can better understand the biological pump and how it'll change in the future. But I sort of want to turn that around and think about what we can learn either from the models or from the process studies we're doing that can give us information about how we should be monitoring for the changes that we expect might be coming in the future, both for sort of the broad scale questions of what we're sort of expecting from the CMIP6 models and what's already in them, and also for some of the things that we think might not be in them, like the feedback mechanism that Jessica was talking about, or building on Melissa's question, thinking about how we might expect CDR to fall over top of what we're looking at. And I guess I could frame this both as what are the kinds of things we need to be observing to be able to have a long-term record to, say, answer Bob's question 20 years from now. And then also sort of what do we know about the spatial variability to say, given the limited set of observations we're able to make, like where should we be focusing those observations to be able to see where those changes will start to emerge first?
That's a very good and big question. I'm just going to address one little piece of it. I just want to give a plug for sustaining our time series. And maybe that's a little self-serving because I'm involved in a lot of them, but I just, you know, we have some really excellent biogeochemical time series, and, you know, the community gets a lot from them, and we just need to be careful about sustaining them and using them, mining the data from them. And so that's just one point I want to make. We have a lot of the information now that will help us with this, and we just, we need to either use it and just keep it going into the future so we can see these changes. So, one comment.
Oh yeah.
Awesome. Just one comment. I mean, if you kind of, you know, one possibility thinking forward, you know, we need a observational system that will look at the state of the system as it's moving forward. We have data assimilation tools today, and so we need tools that will take what we're getting out of BioArgo, or what we're getting from all the surface measurements that we saw yesterday for the PCO2, and assimilate them so we can kind of get a state of the system sort of assessment as what's going on in the biological pump and what's going on for both of the pumps, probably simultaneously. So myself and Tim DeVries and Ivana and Kelsey Bisson have an annual review paper that's coming out shortly, if I ever finish my responses to the And in that, we do kind of try to lay out a vision as to, you know, with these sort of inverse models, can we actually look at the changes of the export and the sequestration numbers as we go forward? We know that the parameterizations that are in those are imperfect, but we have things to drive those solutions to better places. So in some sense, part of that future entails having good external data sets, what we know about zooplankton distributions, what we know about oxygen, what can we say about nutrients, probably particle distributions, large particle distributions, and to assimilate them going forward so we have a sort of this observational record that, you know, this observed model thing, whatever you want to call it, this assimilated data product, that would be able to let us make some informed decisions about doing things like CDR and that sort of thing.
Can I comment on that that question from Hillary? Absolutely, 100% what Dave said. Like, we need better observations and model kind of integration efforts. But I would also say that there's huge parts of the Southern Hemisphere that, I mean, we haven't observed very well at all. And one of the things that I'm particularly excited about that's potentially coming through, and I don't know how much adoption it will get, is the like smaller cameras that can go on the biogeochemical floats, like the UVP6. And I think that will take us a significant way to studying these areas that are very difficult to study.
Oh, green. Green means go. You know, I was going to emphasize a previous kind of comment that you made during the session, which, you know, there's only so much oxygen in the sea, right? And it gets to this point you made about when we combine the new biological measurements that we've been talking about, the mechanisms with these fundamental biogeochemical constraints that are quite powerful. Oxygen in the ocean, nitrate in the ocean. When Jessica, in her work, when we first started adding in this new paradigm of these important salps, the first question is, can you reconcile that new paradigm with all those biogeochemical constraints that you know about the ocean? So I think that casts a more positive light on our data constraints, because when you combine this new information with those biogeochemical constraints, we actually know quite a bit. And even the models that Charlotte shared earlier, there are some of those that we know are wrong. There's a big spread there, but you look carefully at some of those and you can say, okay, well, that model has a process that needs some work. And so, maybe that's a more, the point that you made, David, I think is a more bringing together all those constraints will move us forward. It wasn't a question because you answered it, so Hillary beat me to it.
Oh, well, that was efficient for once.
Thanks for waiting. In the models of future oceans warming and how that changes its flux, you kind of change the remineralization rate. But many people are working on warmer oceans, selecting for smaller organisms, smaller fish, smaller zooplankton that are going to have different functional relationships. Debbie showed the trait-based models. And so when you mention oxygen, many of these animals are living just barely getting enough oxygen. So when you raise the temperature, you decrease solubility and increase the respiratory demand. So Nikki talked about that in the double whammy yesterday. So we're already seeing changes in the size spectrum of zooplankton and fish in coastal communities that are warming a little faster. And so in the open ocean, if this happens, would logically have implications on the flux. So model that.
Michael, you can only, you need to model what you can see, right? That's as we move into environments, it's going to be incredibly challenging. I mean, that's one of the reasons I think that having some sort of, integrated, data model systems that let us understand this is going to be super important.
So we must move on to the next thing in our agenda, but it requires more discussing this topic. So don't worry, those of you that still had hands. Okay, so we've blown up our schedule because we've just had such a lovely time talking, which is great and is the whole point of today. But we really want to make sure that we're having these small breakout discussions so that we can split up, think about questions, think about things that we've talked about today. These are some topics that we as a group, the speakers, the organizers have kind of brainstormed before coming here to OCB, and it's rewarding because many of these things have been discussed a little bit today.
Okay, so let's start talking as people are coming in. So I really have no idea how to do this, because if your table was same as mine, we jump through many different topics and solve the problem of biological carbon pump forever. But what I'm going to do, if it's okay, the leads of the discussions on different tables, which are the presenters today and other unfortunate souls, they were listed there. If you guys can kind of like come closer, and if you have some notes and stuff, and I think it would be best that we start with maybe whoever was first. I think Susanne was first. So I don't know if you can just say one topic that was the prime topic on your table, and let's start from there, and then. And I can start from mine. Do you want me to start? Yeah, so like we were really like technology-oriented table, and we made a shopping list of the things that we want. And I can scroll down, but in theory, we made like a really long list of instruments that we need. And it was size fractionated primary productivity in situ, in situ DOC, in situ respiration, and so on and so on. If you actually jump on that Google Documents, you're going to see that there's lots of need for these continuous measurements, in situ continuous measurements of the rates and things that we're missing, and also higher trophic levels. So like, exploring some kind of acoustic sensors that we can minimize and put it on sail drone, as Jess's group is doing. But then we kind of continued discussion, and I raised the question that really bothers me a lot. Like, every generation has a fab. When I was a grad student, we had a fab of gliders, so everybody had a glider. And the majority of those gliders are now sitting on a shelf. Every single glider that I worked with in my life, except for Craig Lee's gliders, are sitting on a shelf. The problem is we're entering this stage where we're collecting all this imaging data and stuff. It takes a long time to process that data. It takes a lot knowledge, and everybody usually gets funded to get an IFCB. And then me on a NASA validation side, I'm waiting for the IFCB data to be submitted. So, like, what can we do as a community maybe to not just, start thinking about developing of the instrumentation that we need, but also try to find a better way to seize the instrumentation data that we're currently collecting, because we're obviously not making it available for everybody. Does that make sense? Kind of like, maybe not so much talk, maybe, yes, we need instrumentation, but also we need like processing capabilities, funding for people who are going to process that data. And also means to get that data available so we don't have fights where, I'm going to look at Mike, he's my favorite modeler, make it easier for modelers to find it so they can, change their things around. So like, Jessica doesn't have to go and, dig through everything. So it's not Michael only here. So I don't know if any other groups talked about instrumentation, similar problems. I don't know. Group leads? Anybody nobody talked about instrumentation? You guys are happy? Yeah. Okay, yeah. Okay, I have Suzanne, Jessica.
So we also had a meandering discussion, but one thing we came away with as a field, we have to be bolder and more confident in demanding that we get the resources so we can ask these important questions and answer the important questions. So we need a latitudinal, seasonal experts, for example. And one comparison we made was to all the resources going into economic models, weather models, and in comparison, our field is much smaller and much less well funded. So to really strengthen our outreach in terms of what people are standing to gain from what we can understand by better studying the ocean.
So, if I'm understanding correctly, so like put it in monetary value on our science in a sense? Is that what you're saying? I can move this down. I have it.
Sorry, not monetary value,
I have
but
my imaginary glass of wine, like I was yesterday walking around with.
Not the monetary value, but linking the unknowns to consequences.
Yeah, that makes sense, yeah. Anybody else? Okay, Dave has something to say. Yeah, but can you come here? Every time I get a microphone, people take it away. I don't want to give it away.
Oh, shoot. Okay, good. Hey.
Hey.
I was in a bad bed.
We did do all the time.
So we talked about instrumentation, and we just sort of, we went down another path. We just sort of listed the things that we wish we could have and wish we could have better. And the carbonate system came up again, being able to measure reliably alkalinity in DIC. We talked about DOC and how we don't have sensors to measure DOC. Am I repeating what you just said?
No, but these are the same. I just kind of went, I can scroll down a little bit.
Yeah, and especially DOC reactivity. I mean, or bioreactivity. How do we know that we're actually getting the relay all fractions? We talked about eDNA some we talked about DNA in a bunch of ways, including improvements to the ESP, which I don't even know what it stands for anymore. Environmental sample processor. Hey. And the possibility of doing sequencing on a chip, maybe for specific things. One desire was to have much more automated filter collection for genomics, so we cleanly collect things, put it into libraries. We did talk about DNA collections in libraries, kind of like we do cores. So we have sort of more of a facility where we can share from data from data experiment to data experiment. So in some sense that investment that the national governments are making in us to collect data sets can be used by other people as methods, especially genomic methods, get more and more useful. And that's all we talked about. Table 2 out.
Yeah. I don't want to give mine away. This is the only thing I'm getting.
I'm going to jump in because we put our stuff down under leveraging existing data, but it's very similar. So our table 17 to 18 talked about model observation intercomparison and or integration. Basically, the question was, how do we better bring observations to inform models or models to inform observations? And overall, we talked about so many different things, but there's basic lack of uniformity in data sets. One of the people in our group talked about how there's a really lack of data sharing in the Indian Ocean and validation of both oxygen and chlorophyll data. That there's lots of efforts to fill in data gaps, but also it's hard to, as a modeler, to get those data. There was a comment, someone said it was very painful to reprocess data sets so they can be compared to models. And there needs to be kind of this group of folks who are just doing data curation, and it's not very sexy or cool, but it's super necessary because modelers can't deal with the data and the complexity that data are usually uploaded to repositories. We talked a little bit about phytoplankton community composition, about the possibilities of hyperspectral data, how exciting that was. And then we finished up by saying that there was really a lack of training in open data skills and data science techniques and all of this kind of at the graduate level, that we need more training for scientific computing and data wrangling within our whole community. Somebody said NASA products are very well organized, so
I can agree to that. No, but I mean, we can go to the next table, but I think this kind of in a sense speaks to what I've just said, I mean, at least in my head, we have this data. I mean, and I can say, like, I work sometimes in the validation side, so I know your dirty laundry. So, everybody who's funded under NASA has to give stuff to us. And the dirty laundry goes to lots of washing. Like now we really have implemented quality controls and stuff, so I know everybody's dirty laundry. And there's a thing, if you're saying that the models are not replicating stuff, if you give junk to us and we don't have a way to control and actually put human power to control the quality of the data, that's what the modelers are going to be picking up. Or they're not going to be picked up because we're not going to put that stuff out because it's bad data. So there has to be a connection. I know that PICO-DEMO is also trying to do a fantastic job in curating data and actually making it available. So there's responsibilities from all sides, but also as we're collecting more data, we have to think about how to make the data available for the modelers. With hyperspectral radiometry coming along the way, we have, once again, IFCB. How do you put that in a database? Who has put IFCB in a data? I mean, who of you here has done, everybody's doing phytoplankton structure. Has anybody put actually phytoplankton data in any databases? Exactly. So we're not going to be able to validate, oh yeah, okay, I'm sorry, you have. But, in a meaningful format that can be picked up by modelers, that can be picked up by me I'm sorry for the few individuals, but I just want to point out it's a hard process. Sorry. So anyways, other tables?
Yeah. Either way. Okay, well then, tables 7 and 8 reporting here. We actually talked about many things that Jessica also just mentioned. Basically the same story, that as a modeler, it's hard to use data of observation lists because it's often painful, was the word, very difficult to find. It's scattered in different papers, and with a lot of work collecting it, often we are then not probably just trained enough to know how exactly to interpret it. And I just wanted to add that I see that it's a lot of work to do so, and we probably need some champions, heroes, to create those databases. But I feel there's also a lot of in it for the people who do it, because, I mean, these databases usually get tons of citations once they're out there. So it's not that it's only hard work with no payoff. What else do we have? Right, we also talked about model intercomparison, and the point was made that, for instance, for many of the models, the Earth system models that I've showed today, it's
often not really clear which of them are actually better or worse in what areas, and how much you can trust them in which regions or timescales or whatever. And maybe more effort should be put into model comparison also. And then we had a point, the last point, also about instrument development. And there I don't feel qualified.
Yeah, we have had an interesting discussion on, like, for instance, developing fleet of drones to resolve, like, phenomenon that are not that well constrained with satellite data due to cloud issues or low temporal resolution, while fleet of drones could be like provide like high temporal resolution measurements and go around, like better resolve like coastal processes and this go around these sorts of problem. And then everyone agreed that the talk of Colleen this morning like pointed out a lot of super interesting new technologies to analyze, to study the deep ocean, like the flux of particles. This is going to be very important to better constrain the transfer efficiency. And so any further development which will allow to deploy these type of instruments for longer time, like so in order to have high temporal resolution of data, is going to be very helpful to improve like estimate of flux attenuation, type of particles, and I will also add the interaction between particles and the zooplankton and how this could change with the changing ocean. And all those like sounded really promising. So any effort towards improving those is going to be helpful.
Also, thank you so much to everybody at my table. That was a very nice discussion.
Okay, well, I can report out from tables 9 and 10, and we have it kind of similar. We started off being motivated by this question, how do we make the models find the right answers for the right reasons, and kind of settled on we need more sustained high-resolution observations. I mean, I probably biased that answer, but also not just like you were saying, Ivana, like not just more instruments. We need more instruments in the water, observing this in a sustained way, but we also need to invest in the people power, especially to actually analyze that data. And I'll just add my own thing in here. I think trying to find meaningful ways to bring in the data science community and the computer science community, and that's a hard thing to do because, they can get paid a lot doing other things. It's hard to compete. But I do think they are already solving these problems easily, and we need to bring those people in somehow. And then, we're saying, how do we figure out the best place to put these observations? Is there a way we can be strategic about that? If you wanted to start a new time series, so we're talking about, time series, and also how do we have time series that don't rely on heroes, right? A hero that just keeps it going, because that's not it's a pretty vulnerable way to have a time series. And so it's that was basically what we talked about.
All right, I'm just going to jump into this. One of the comments that we had in our table about this time series, how this time series is already existing, and we have new instrumentation. Maybe that's the place we want to deploy it. I mean, like, try to make this push and find money to actually, adopt the novel instrumentation. I mean, we've been using that stuff, but add UEP to all these long-time data series, and actually, show support for the processing. Not just add a UEP and then somebody will deal with data. Add this new technology to the, long-time data series. Keep the climate record going, whatever you were, because that can then inform the stuff that was happening in vacuum. It's a place to develop proxies now. It's a place to develop really meaningful relationships that can be then used in models, but also used for other campaigns. And then not just buy UEP, pay for the person who's going to deal with it, pay for the software person, make sure the data gets submitted. So, you want to go? Sorry, I'm just jumping around.
Yeah, I'm reporting from tables 19 and 20. We didn't really go in the categories. It was kind of all over the place, and we came up with probably with more questions than answers, but it's going, again, down to the models and the empirical studies that
just through the talks today, the biopump is way more complex than we originally thought, and the new sources and mechanistic relationships are highlighting strengths and weaknesses of both models and empirical studies. So the questions that came from that conversation were, what components and processes are most important for modelers and empirical studies to tackle together? What metrics do empirical studies need to capture to improve or better constrain the models? And how do we best capture temporal, spatial, community composition dynamics in field studies that are useful to the models? So that was kind of that conversation for that part. And then another takeaway, we talked mostly about kind of the things that stuck out from the talks today, was about SELPs and how they can change the size and direction of the feedback, and that simulations of current, especially those with small phytoplankton-dominated systems, and especially future flux don't incorporate that component, except for Jessica's model now. So quantifying specific trophic interactions and community composition seems to be a very important thing to pay attention to. And then there was a question about, will this be a similar situation with the rise of jellies, or are SELPs very just unique in that respect? So that was kind of a question that came out of that, thinking in terms of like long-term impacts. We had a question about DOC labelity dynamics. So how does recalcitrant dissolved organic carbon affect long-term storage in the ocean? And how might that, how does that play into the biological pump? And then a question about DNA. I think there was more just like amazement about just how ancient DNA can be stored for that long. So that was more of a comment than a question, but there was a lot of surprise on that. And then finally, we need more coastal to open ocean transport investigations to better understand how coastal processes contribute to global carbon export and sequestration. That was our discussion.
Sure. Tables 11 and 12 reporting. So we started mostly talking about this integration across time and space topic, and what were some of the things we'd like to see, and we kind of got on this topic of linking paleo record and the sediments with organic carbon preservation, so using the past as an analog for today, and that there are these relationships between temperature or carbon dioxide level in the air, but how do these relate to organic carbon preservation? And it was brought up that wouldn't it be neat to be able to tie phylogenetics into organic carbon flux and preservation, like do the phylogeny of SELPs or some other important taxonomic group of plankton? When did they diversify? Did they diversify when it got really warm, and did that increase organic carbon preservation in the sediments? And could we probe some of the phylogeny of some of these important groups? And so that was a fun discussion. And we also talked a bit about wish lists for things we'd like to see. We got on that topic for a bit, and some of this should be covered tomorrow, but more autonomous time series on continental shelves and coastal continental shelves in general. What is their potential for carbon storage? We also another wish list for sampling was underway sampling, molecular deep capability for doing underway sampling for deep sequencing. So molecular measurements at high resolution, underway, same way we do underway temperature or chlorophyll, but boy, wouldn't it be cool to be able to do that underway rather than stopping and taking samples, like Terra Ocean's data, which is amazing, but wow, what if you could do it underway? So that was a wish list item. What am I missing? In situ DIC was on the wish list. And that would be great, huh? Did I miss anything, group? Okay.
Anybody? Great. Thanks. So just a super quick report out for one more group, and then we're going to go to a report out for the online breakout that has been going on. Our group talked about a lot of the things that have been mentioned, so I'm not going to duplicate any of those. One thing I haven't heard yet is we've talked in the last couple of days about the value of the growing BGC Argo array and the ability of it to provide like global coverage and sustained observations of the BCP potentially, and so doing some process studies that really bring in the BGC floats to in-situ observations so that we can really kind of understand the measurements we're getting from the floats in order to kind of scale them up for bigger scale picture questions will be important. And then I'll leave you with a philosophical thought that somebody in our group had, was thinking about the way that historically examining the BCP has been really geared towards looking at lower trophic species, and so we have this lack of long-term observations, I mean even for lower trophic species, but especially for upper trophic species, and thinking about fish today and their importance to the system. We had this thought about in terms of long-term changes that could have occurred to the BCP with fishing and exploitation of our oceans, and how maybe there was some compensatory changes in the system, and just how impactful has fishing been on this like fish component of the BCP, and how has that potentially changed over time, and just thought that was really interesting.
I know we're going to the next group, and you can answer the question. We have to go to online, and you can ask a question if possible. Can I ask all the leads to like copy paste stuff into this document? Yeah, she's setting us up.
So, no, you know, just there was one more thing that we talked about. I don't want to repeat what was said before, but there was a discussion about kind of figuring out ways to simplifying the path for innovation in modeling. And so we were talking about thinking about making sandboxes or some sort of way that we can test ideas, run it against a model, beat on it, you know, that sort of thing. It could be done in a regional way, you know. But this idea of a sandbox, we can test models, we can test new hypotheses, run it against constraints. That sort of came up, and I didn't hear that in the discussion.
So I'll be reporting out for the online group. I assume you all can hear me. Dave's comment actually pretty much mirrored what we got to in the end, which was, so we focused on the integration across spatial and temporal scales, because you heard an awful lot today about you know, the importance of these ephemeral events and how complicated they can be. And right now it seems like there's a bit of a disconnect between the process level and mechanistic understanding and our ability to improve our estimates of large-scale fluxes that we have from either satellites and statistical relationships or large-scale mass balances. And so it seems like exports is doing a good job with this, but you know, integrating observations so we can match observations of ecosystem dynamics, stocks, rates with the integrated chemical changes, so that we have testable hypotheses that can be probably put into regional modeling studies before we try and look at large-scale Earth system changes. So we can see whether or not we're actually deriving mechanistic understanding in a way that we can use to improve our predictions. But obviously, as Charlotte said earlier, and as has come up a lot, the questions you're asking matters. So short-term and long-term questions might have different levels of mechanistic understanding that you need for small scale, large scale. And so, you know, if you're talking about paleo level, looking at organic carbon burial on thousands of years versus short-term perturbations or seasonal cycles, all of those will have different levels of understanding. But really, it seems like people are doing the sort of contemporaneous measurements that need to be done, but continuing to work on the exchange of ideas and data and analyses between, say, the self observationalists and biogeochemical Argo folks and modelers is really seems key right now, so that we're taking what everyone's learning at the really small scales and able to extract out the relevant levels of mechanistic understanding.
it shouldn't be me. It should be one of you too. It should be me. Okay. So, well, I mean, this was really kind of done really fast, and we like just kind of blurbed out. But there's lots of overlap, and there's, you know, I think we all see the similar problems and have similar wishes and stuff. So, if the leads can just plop that stuff in a document, I think the idea from the two lovely ladies who organized a fantastic session. And I have to say something that was pointed out by Tammy there, that it was a really beautiful thing to see 8 ladies of the 10 panelists. And that's something that is, I mean, I just feel like, you know, would have to point out that stuff. And that's, you know, that's thanks to these lovely ladies who have organized it. But, I mean, just, yeah, put the rest of your comments in the document, and I think they're planning to do something magical with it, and yet to be determined. But on behalf of you guys, thank you so much. This was an awesome day, great discussions and everything. Why am I doing this? You guys already.
Yeah, so I think we've had a long day. It's 5 o'clock, and we want to make sure we get you guys out of here. Thank you so much for loaning your brain trust to this topic today. I think we're not sure what we're going to do with this information, but we have big dreams. And just thank you so much for your time. Thank you so much to our speakers. This was such a cool session to work with you all on, and have a lovely evening.